Hi, everyone. I'm Teresa Strong, Director of the Research Program at FPWS. Before we begin, I'd like to thank all of our today's conference: Soleno Therapeutics, Levo Therapeutics, Harmony Biosciences, Radius Health, Inversago Pharma, Saniona, Consonance Therapeutics, and Novo Nordisk. We really appreciate the support of these organizations in facilitating sharing of some really fascinating scientific information that we'll hear over the next couple of days. And we really appreciate all of you joining us. Of course, we wish we could be in person, and we will be next year. We're excited about that, but we appreciate you joining us virtually today. We have a brief welcome from John Walter, FPWR CEO.
Hello, my name is John Walter. I'm the Chief Executive for the Foundation for Prader-Willi Research. I'd like to welcome all of you here today for our 2021 Research Symposium. We have a great lineup of speakers for you today. We are looking forward to hearing feedback from both Levo and Soleno on their completed phase 3 trials and the work that's taking place to hopefully push them over the finish line with the FDA in the near term. We'll do the fingers crossed on that one. We also have great speakers in various stages of research that we'll be presenting today. I'd be remiss if I didn't remind you to go to our to our YouTube channel and look at the abstracts. We have a great group of abstracts that have been submitted for review. So I'd encourage you to do that at your breaks or before the sessions begin. Later today, we have a breakout group where we'll have both a genetic and molecular track as well as a clinical track. And so I'd encourage you at break to take a look at that and see which ones you want to sign up for and participate and listen in on. Finally, I'd be remiss if I didn't thank our generous sponsors that have helped make this event possible and bring all this information to you. I would also encourage you to provide feedback or ask questions to any of us, either during the presentations or after the presentations if you'd like follow-up. God willing, we're going to be in person next year. I'm looking forward to that. I think we're all pretty much tired of Zoom and or Teams or whatever platform you're working on, and it'll be great to be back together with all of you again next year in Chicago around the same time of year. With that, I'll leave you to the conference. I hope you enjoy it and look forward to hearing your feedback. Thank you.
So yeah, as John mentioned, our conference is set for next year in Chicago. So please mark your calendars for the same dates, actually, September 29th and 30th. A few reminders and housekeeping. So please remember to take a look at the posters. If you scroll down to the bottom of the landing page, you'll see two buttons. One will take you to the actual posters, so the PDFs of the posters, and another will take you to our YouTube channel. We had so many excellent abstracts submitted this year that we could not give everybody a full oral presentation, but what we asked people to do is record kind of a walkthrough, pretty similar to what would happen in an in-person poster session. And so we've put all of those up. So they're just, real brief, three or 4 minutes about each poster, and you can scroll through those. So there's two buttons there. For the oral presentations today and tomorrow, we will have about a 15-minute presentation and then about 5 minutes for questions. We welcome your questions. You can just add those to the chat box during the presentation, and then I will be reading those to our speakers, and they'll be answering. And we'll encourage our speakers, if we are not able to get to all of the questions during the oral session, to respond to the chat as well. If you have any technical problems, we hope that will not be an issue, but I'm told that the easiest thing to do is just try refreshing the page, and that usually fixes most things. There's also a link at the bottom of the page for a alternative live stream if that becomes necessary, but I think we should be good to go. So with that, I'm really pleased to open the conference with a talk from Elizabeth Roof from Vanderbilt University. She's talking on behalf of Levo Therapeutics, and she's going to tell us about intranasal carbetocin, reducing hyperphagia and behavioral distress in PWS. This is the results of the CARE PWS phase 3 study.
Hi, my name is Elizabeth Roof from Vanderbilt University, and I'm going to be talking about the CARE PWS Phase 3 study results today. Just so you know, I am a clinical advisor for Levo Therapeutics. I'm also a research sub-investigator for the CARE PWS study. The CARE PWS study is sponsored by Levo Therapeutics. I'm also a research sub-investigator for the Harmony study and also a research sub-investigator for several previous clinical trials, including Zaftion, Rhythm, Fairing, and Melindo. And I also have gotten some previous research funding from PWSA, FPWR, and NICHD, just so you know. So I'm here today to actually talk about oxytocin and carbetocin. And so there's a really long-standing interest in oxytocin in the PWS community because it's a hormone associated with many of the physiological features of PWS and is involved in appetite, anxiety, and social and emotional functioning. People with PWS seem to really fit this model of having some aberrant kinds of behaviors that are related to oxytocin. Aberrant levels of oxytocin are implicated in both the Schwab and the Bachakova studies, showing fewer oxytocin neurons and expression. If you look at the two slides, you can see the one with controls have a lot more green, bright green, which shows you those oxytocin neurons. And then if you look in the ones with PWS, way fewer neurons, way fewer expression. And so again, it's interesting to see that physiologically there are definitely some differences there. The Einfeld study in PWS showed that increased doses of oxytocin could increase aggression outburst, possibly due to off-target effects. So again, there's some interesting physiological things, which makes us very interested in oxytocin and how it actually relates to PWS. Enter carbetocin. It's an analog of oxytocin and that shows some key differences: a longer half-life and improved selectivity of target effects affecting vasopressin. IV carbetocin is approved in 90 countries outside of the U.S. for excessive bleeding following a C-section. A previous phase 2 trial in carbetocin demonstrated significant improvements in both hyperphagia and OCD symptoms in PWS using only the 9.6 dose as a means to show signal. So again, there's some interesting data that shows that carbetocin is relatively safe. It's been used many, many times in a different preparation, and the phase 2 trial showed some significant results. Phase 3 looked at two different doses, 9.6 milligrams, which was used in the previous phase 2 trial, and the 3.2 milligrams, both given 3 times a day to about 175 participants, children aged 7 to 18 at 24 different sites. Most of those sites were in the U.S., a few in Canada, and one in Australia. So again, you're looking at an 8-week placebo-controlled period with everyone receiving active carbetocin after that for a 56-week long-term follow-up period. So again, at that point, people were actually allowed to even go on to a long-term extension period where they continue to get medicine. Several of the folks that we saw in the trial continue to get medicine to this day. So there was a 1-to-1-to-1 double-blind period that lasted for 8 weeks, followed by that 56-week extension. All of those on placebo who were randomly assigned to either the 3.2 milligram dose or the 9.6 milligram dose at week 8. So again, you can kind of see the schematics of how the trial started, what the changeover was, and at that point, for the 56 weeks, everyone was on a 3.2 milligram dose or a 9.6 milligram dose. Then COVID hit. It had a huge impact on everyone. The study held new screening on March 12, 2020, and then the study closed to screening on May 30th, 2020, resulting in 130 participants with PWS instead of the 175. The FDA agreed to Levo's proposal to conduct a pre-specified primary efficacy analysis before unblinding based on data collected through March 1st, 2020, basically when COVID hit. This data is named the primary analysis set and it has 119 patients in it who actually were in the trial, and we're going to talk about that. Here you can see that the primary data set includes almost all of those randomized, and that the 3.2 milligram and the 9.6 milligram cohorts are evenly matched as far as size. There were a group of people that were randomized and not dosed, a very small number of those people, often because of issues with not actually meeting qualifications at that visit, even though they were previously randomized. So those are in there as well. Here we're going to take a closer look at this cohort. You can see that the evenly matched groups with similar baseline scores for hyperphagia, OCD behaviors, and anxiety. The age, weight, BMI, genetic subtypes, again, you can see across all the different treatment arms and placebo. And then you can look at those baseline scores of hyperphagia questionnaire, the CY box, and then the PADQ. And so again, you can look at those to see how those groups compare, and they're incredibly similar. Randomization across the three arms was equal. Given that the 9.6 milligram dose was the only dose used in the phase 2 trial, it obviously became the primary endpoint, and the 3.2 milligram dose was secondary endpoint. So again, you can look at the efficacy measures and the fact that the HQCT was the primary endpoint, and the Children's Yale Brown Obsessive Compulsive Scale were also an endpoint, trying to look at whether obsessive compulsive features change in people with PWS. You can see that the data shows improvement on hyperphagia for both doses over the 8-week period. So again, the gray is the placebo arm, the orange is the 3.2 milligram dose, the blue is the 9.6 milligram dose, and the yellow bar is a combined group of 3.2 and the 9.6 milligram doses put together. So again, you can see that those differences across hyperphagia in that baseline to week 8 group. Here you can see that the data shows improvement on the hyperphagia questionnaire, the PADQ, the clinical global impression scales of both change and severity, and trending improvements on the SIBOX. So again, you can look at the 3.2 milligram dose, the 9.6 milligram dose, and then the combined group. So again, it across the board showed improvement, except just trending improvement on the Y-box. Here you can see that the improvements were quite durable over time, even through a pandemic. So for those that carbetocin was effective, those effects were clinically meaningful and sustained over time. So what's interesting is you looked at that earlier slide, and you can see that these continue to go down. And again, there's definitely change. So the light orange bar is placebo to 3.2 milligram dose. The dark orange bar is just basically they started on 3.2 milligram dose and continued on that. Light blue was placebo to 9.6 milligram dose, and the dark blue bar is actually on 9.6 at the beginning and stayed on 9.6. Here you can see that for the 130 participants, the mean hyperphagia score was around 23. The hyperphagia questionnaire was one that we developed and actually published in 2007, and this is a revised version that was been validated in several clinical trials, including this one. And a max score is 36 and a minimum score is 0, and there are 9 items that are scored 0, which is none, all the way through extremely severe or all the time kind of thing. So basically it gives us an idea of severity across a number of these domains. So here you see that the improvement in hyperphagia on both treatment arms, with the 3.2 milligram dose showing the most improvement. Across the SIBOX with a mean of 26 at baseline, and across the 64 weeks trending towards improvements over time. So again, this is the one that didn't have significant change, but you can see that there are changes across the board for the both the 9.6 and the 3.2, with again the 3.2 milligram dose showing the most improvement. Across the PADQ, improvements were seen across the board and sustained over time, again with the 3.2 milligram dose showing the most improvement. I think it's important to look at this point that we're actually well into May 20th of 2021, and you can see that those effects are holding over time. I know for us, we were incredibly pleased to see that people started out on medication or placebo, and at week 8 quickly switched over to active medication, and that the effects were seen and sustained over time. I think it's really important for us to kind of look at this idea of being able to show that the drug works long-term. It's not a short-term solution. It seems to be a long-term solution over time. So I think that for us was the most compelling part of this data. So here you can actually see that the 2 placebo-controlled studies demonstrate that intranasal carbetocin effectively reduces symptoms of PWS. While the 3.2 milligram dose was not previously chosen as the primary endpoint, it nonetheless achieved significant reductions in hyperphagia, again at a p-value of 0.016. There's an internally consistent effect observed with the 3.2 milligram dose across multiple measures, in addition to the HQ-CT, including the PADQ at a p-value of 0.027, the CGI clinician impression at a p-value of 0.027, and the CGI severity scale at a p-value of 0.021. A strong signal is also seen when pooling the active doses. So here we have a p-value of 0.055 for the HQ-CT and a p-value of 0.035 for the clinician's kind of global impression of severity. So that was, again, pretty impressive. These improvements in PWS symptoms are sustained over the 64-week period across both doses. Now I want to focus on the safety data. Let's look at the safety and side effects. You can see here that the placebo-controlled period, the drug appears safe, well tolerated, with similar mild side effect profiles across all treatment arms. Even people on placebo showed some side effects, right? And so it helps us to know that they are basically, the drug is well tolerated, it appears to be safe, and again, nothing really significant as far as any huge issues here. Over a longer period, the safety data shows that the drug appears, again, to be safe and well tolerated, and patients didn't discontinue due to these side effects. And so here we're looking at side effects as common as headache and flushing, which are fairly common with this drug given the vasodilation properties that it has. But again, things like fever and everything else could often be related to just illnesses that children get. So again, those things are all included in the adverse event kind of profile, but helps us figure out that across the board, again, safe, well tolerated, and people did not drop out because of that. Additional safety data, including clinical labs, ECGs, and vital signs, were monitored throughout the study. So again, at every visit, these things were included. The sponsor and the DMC, the Data Safety Monitoring Board, reviews of additional safety data have been identified that there were no notable trends or apparent safety signals. D-dimer results have shown no apparent trend for a change from baseline in either treatment arm, and no events of deep vein thrombosis or pulmonary embolism have been encountered to date. So I think it's important, given the past history of some other trials, that out of an abundance of caution, D-dimer was actually looked at, and again, no apparent trend for change in the baseline or the treatment arm. They also monitored serum sodium, which has also shown no apparent trend from change to baseline in either treatment arm. So again, if sodium were to actually be lowered or depleted, that could be problematic. And that was obviously a concern with oxytocin because of those vasopressin effects. But in this case, we don't have any issues with that. It again shows it to be incredibly safe. So overall, what I think this shows is intranasal carbetocin reduces hyperphagia and anxiety behaviors in PWS with maintenance of improvements over time. The data support a highly favorable overall safety and tolerability profile in participants with PWS. LEBO and the Independent Data Monitoring Committee have agreed that the aggregate data support continued advancement of the 3.2 milligram dose as the lowest effective dose, and LEBO is working closely with regulatory authorities to bring LV-101 to patients as quickly as possible. And I know for the 20 some odd patients that we continue to see in this trial, we're very excited about the possibility this drug could actually be something that would be available to many families and actually help relieve the symptoms of PWS. Thank you so much for allowing me to talk with you today about the carbetocin trial data, and I'm happy to answer any questions that you may have. Thank you.
Great. Thank you so much, Elizabeth. We appreciate that talk. We do have a couple of questions that have come in. Again, as a reminder, if you have a question, please just go ahead and put it in the chat, and then I'll ask the presenter. So the first question was from Larry Ryder, and he was asking about why would the 3.2 milligram placebo continue to improve over time? I'm wondering if that was maybe a misunderstanding or terminology, because I believe it was you were transitioning from placebo to 3.2. Is that?
Yeah, no, that's actually a great question, Larry. So we're not really sure, but my thought is having watched all those patients, I think this idea that when you're taking nothing, you see a much bigger kind of like, oh, look, I'm better kind of thing, that maybe that was actually kind of what showed up. But we really don't know. I think it's really fascinating to see what the placebo effects are in people with PWS. Over many trials, we've had people say they're better even when we don't see any evidence of that. So again, I think it's something that we don't know. And it's not a big change, but again, that 3.2 milligram dose really seems to be where the money is as far as effectiveness and seems to work over time.
And Larry was also asking about, how do the improvements break down by genotype? I guess I would add to that by age. Are there particular subgroups that seem to respond better than others?
I don't know that. I haven't, as far as, been unblinded since we're a site, but my thought is, it seems like across the board, people generally did well. Like we didn't see a lot of that within the 25 or so people we had at our site. It seemed to be really effective. And, we know that adults have some of the same symptoms as kids, right? So there's a real thought that it could be effective for adults as well. Don't know that, but feel that some of our folks have aged into the adult range and seem to be on drug and doing well. So thoughts that we hope that, it seems to be effective for a lot of people.
Great. And Sarah Fagan says, thank you for an interesting talk, and asks about what is the relationship between reduction in hyperphagia and reduction in anxiety. Did that seem to go hand in hand or differences?
The $1 million question, right? So we don't know. It seems like for some kids, the anxiety seems to really be part of hyperphagia. They're anxious about food. They're anxious about a lot of things related to food, including schedules. And then for other folks, they don't seem to have, anxiety seems to be really separate from hyperphagia. So I would say that it doesn't seem to be a, strong correlation for everyone, but I think it's really more individually based. So we've never really figured that out, but we do know that there are people who aren't even hyperphagic who already show significant anxiety. So for us, we think that they may be related sometimes, but other times they may be related to other kind of like psychiatric and more kinds of like schedules, expectations, worries about things not being consistent, and really have not much to do with food. So just depends on the kid.
Great. Thanks. And Elizabeth Roeder asks about if approved by the FDA, what would be the age range for treatment?
That's not something I can answer. I would assume for me that if it's approved in children, obviously that would be the age range it would be targeted. But I mean, there's probably nothing to suggest that if adults couldn't benefit as well. So I'm not sure. I'm assuming the FDA will let us know all that information soon. So again, it's something to definitely be, kind of looking out for. But I'm hopeful that adults could actually benefit as well, just because we feel that they have a lot of the same symptoms and it doesn't go away, right? They are still having problems with hyperphagia and anxiety, even as an adult. So I'm hopeful that it will be something that adults could get as well.
Great, great. And again, if anybody has any additional questions, you can go ahead and put them in the chat. And even if you think of one afterwards, maybe the presenters can respond in the chat as needed.
Absolutely.
Elizabeth, I had one more question about the Psy box and the responses weren't significant. Do you feel like that is an instrument problem, that is a drug problem, or what are your thoughts on that?
Yeah, that's actually a great question, Teresa. I truly think it's an instrument problem, right? It's not developed for people with intellectual disabilities and certainly not developed for people with PWS. So their OCD symptoms look a lot more like repetitive kind of behaviors versus true OCD. I don't wash my hands 100 times a day because I'm worried about germs. It's more like I do things over and over. Things have to be just right, or I feel the need to do things a certain way or in a certain order. Doesn't seem to have a lot of obsessional kind of qualities behind it. So I think it's probably just an instrument pick. But again, it was effective in the first trial, the phase 2 trial. And I think, when you got something that works, you keep using it until you find out it doesn't.
Yeah. All right. Well, great. Thank you so much, Elizabeth.
Thank you.
So we'll move on with another clinical trial result. Parisa Salehi from Seattle Children's Hospital is going to speak on behalf of Soleno Therapeutics, and she is going to talk about the long-term results with DCCR in the 602 study, which is an open-label extension to Destiny PWS, and comparison of that data with a path for PWS as an external natural history control.
Hi, my name is Parisa Salehi. I'm happy to be here to discuss the long-term results with DCCR from studies, the Destiny PWS study. Specifically, we'll be looking at the open label extension and compare the results with path for PWS, which we're using as an external natural history control. I am presenting on behalf of the 602 investigators as well as the path for PWS investigators. Just a little background about DCCR. DCCR is a extended release formulation of diazoxide, which if you are an endocrinologist, you're very familiar with in the treatment of hyperinsulinism. This is a once daily tablet. It is a potassium ATP channel agonist. Now, the preliminary results which led to Destiny PWS included a pilot study of 13 individuals with PWS, and treatment with DCCR saw a significant reductions in hyperphagia, body fat, increases in lean body mass, and reductions in aggressive behavior. This promising result led to the formulation of the current study. Now, you may be asking, how does DCCR lead to these beneficial effects in individuals with PWS? Well, this is a more complicated answer. There are many different forms. There are many different levels of action for DCCR as the receptors are found throughout. Centrally, we know that the actions of DCCR can reduce hyperphagia as well as improve satiety. Peripherally, we know that actions on the adipocytes decrease fatty acid biosynthesis and increase beta oxidation of fat, resulting in reduced fat mass. And of course, in pancreatic beta cells, we know that it decreases insulin secretion. We're still not quite clear how DCCR affects lean body mass. The 601 or Destiny PWS study is an international multi-center randomized double-blind placebo-controlled study with parallel arms in individuals with PWS. This is a phase 3 study. The 602 is the open label safety extension arm of the study. Again, a little bit of background about Destiny PWS. Subjects had to have genetically confirmed PWS. They had to be over 4 years of age and between 20 and 134 kilograms with moderate to severe hyperphagia, which was defined by having an HQ-CT score greater than or equal to 13. These subjects were then divided into 5 different weight bands, and doses were given targeting a range of 3.3 to 5.8 milligrams per kilogram. These doses were titrated over a period of time up to 6 weeks, and there was a fixed titration to target dose within each weight band. There were no BMI or body fat inclusion criteria, and there was no recommended reduction in energy intake. In regards to the program design, after screening, there was a 2-week run-in, after which there was a randomization 2-to-1 to DCCR as well and placebo. So we had 127 patients or subjects that were able to be randomized into the 13-week double-blind treatment arm, which was 601. 120 subjects completed the study. 7 did terminate early. Of those 120, 115 were enrolled into the open label extension. And as of August 2021, 95 are still enrolled. Patients were randomized based on baseline HQ-CT score as well as growth hormone status. Now, a little bit about PATH for PWS. This is a study conducted by FPWR to advance the understanding of serious medical events in individuals with PWS, and also to understand how PWS-associated behaviors change over time. Recruitment for this study started September 2018, which is similar to the time which recruitment started for Destiny PWS. We anticipate that subjects enrolled in PATH for PWS were going to receive a similar standard of care to those enrolled in the Destiny PWS study. For PATH for PWS, participants, which were the caregivers, completed internet-based surveys once every 6 months. Over 700 subjects have been enrolled in PATH for PWS, and the study is currently ongoing. When we thought about the comparison of the Destiny PWS subjects to those in PATH for PWS, we specifically looked at subjects who were similar to our 602 population as a natural history control. And so for PATH for PWS, the subjects had to be at least 4 years of age, weight 20 to 135 kilograms, and had to have moderate to severe hyperphagia, again defined by HQ-CT score greater than or equal to 13. And they could not be participating in any interventional study. When analysis was done, we specifically looked at data at baseline, 26 and 52 weeks for all subjects. We also did analysis looking at scores pre-COVID as well as post-COVID, using the date March 1st, 2020 as a cutoff, as due to the pandemic, lockdown started for most areas after March 1st, 2020. Subjects were propensity matched, and analysis focused on change in baseline HQ-CT scores, as well as changes in the PWS profile domains and food safe zone domains. The analysis was conducted by an external CRO independent of Soleno, which is the sponsor for Destiny PWS, using a pre-specified statistical analysis plan in order to decrease bias. Just a little bit note about the subject populations we'll be discussing in this analysis. We'll be looking at safety profiles for subjects that were both in 601 and 602 who were dosed with DCCR. We'll be looking at safety profiles for subjects dosed with DCCR in 602 only. We'll also be looking at analysis looking at subjects in 601 and 602 who were defined as being obese based on BMI cutoffs. In our analysis, when we talk about baseline, we talk about baseline as being the visit prior to starting DCCR. So for instance, if you were in the placebo arm of 601, your baseline would be the visit right before you started the DCCR treatment in the 602 arm. Now, a little bit of a note about the impact of COVID-19 on Destiny PWS. There have been a number of publications looking at and discussing the negative impact on children with psychiatric conditions as well as individuals with PWS, and the effects of this on studies that had to be undertaken during the pandemic. We expected that a number of our important endpoints would be affected by the pandemic, including HQ-CT, caregiver, global impression of change, as well as the PWS profile. Therefore, we did do an analysis looking at results prior to the pandemic, specifically pandemic lockdown. A little bit of background on the 601 endpoint results. So when we looked at all of the data pre and post March 2020, there was not a significant change seen in hyperphagia scores which was our primary endpoint after 13 weeks. However, there was an improvement in clinical global impression of improvement at the 13-week mark, as well as change in body fat mass based on DEXA. When we looked at data up to March 1st, 2020, we saw that there was a significant change in the hyperphagia score, as well as improvement in caregiver global impression of change. Therefore, we did find that these discrepancies were probably due to the pandemic. When we looked at behavioral endpoint results for 601, we did find an improvement in a number of different domains prior to the cutoff of March 1st, 2020. And in terms of endocrine and hormonal parameters, we also found promising improvements in acylated ghrelin, leptin, adiponectin, as well as insulin. Now going into the results from 602 and comparing them to PATH for PWS, when we look at the demographics for our subjects, there was a slight predominance of female subjects. It was a young population with an average age of 12.9. The majority of the subjects were recruited from the United States, and about 63% were deletion. The majority of subjects were on growth hormone. For those who weren't on growth hormone, the majority were adults. And as you can see, the majority of subjects completed 13 weeks and went on to complete at least a year of treatment. And as of August, we still had 95 or 82% of subjects enrolled. Now, this is a result that we're very excited about. In terms of our primary endpoint, we did see that the hyperphagia scores decreased over time. And if we look at the trend, we see that as time increased, the change was more significant. And anecdotally, myself, as well as some other of the investigators, can tell you that we found more significant changes in subjects through history from family, as well as behavioral changes as time went on. And as you can see, that the change is more significant after 26 and 39 weeks. This table demonstrates the same concept. At 13 weeks, there was a change of about -6.5, which was a significant change from baseline, but by 52 weeks, the change was -9.9. When we compare this to the PATH for PWS subjects, who are subjects who were not receiving any medical intervention, we found that at 26 weeks, there was a significant difference in the HQ-CT scores, and as well as 52 weeks. And as you can see, the difference in scores is similar, but you can see that at 52 weeks, we had more of a change. Now, we did do an analysis to look at what we considered a reasonable threshold for being a responder. Specifically, if you had a change of score of less than or equal to -7, we found that to be an optimal threshold for having a clinical meaningful response to DCCR. And as you can see from this table, the bar graph, the subjects who were on DCCR had a 60% responder rate, meaning that 60% of subjects had a score of at least -7, and only 20% of PATH for PWS subjects did. When we look at behavioral changes, we also found that those who were receiving DCCR had beneficial changes in PWS-related behaviors, including aggressive behavior, anxiety, rigidity, irritability, compulsivity, depression, and disordered thinking. When we look at body composition results, specifically based on DEXA scan results. We found that those who received DCCR had an increase in lean body mass, which led to a improvement in lean body mass to fat mass ratio. When we did an analysis looking at only those who were considered obese based on BMI, we found that the BMI Z-score also improved. We found similar improvements in endocrine and hormonal parameters after 1 year of being on DCCR, similar to what we saw in the 601 results. So there was a decrease in leptin, increase in adiponectin, decreased fasting insulin, and decreased HOMA-IR. In regards to safety data, the safety profile we found for DCCR was what was expected for diazoxide in general. There were 115 treatment emergent adverse events. 5 of these resulted in premature discontinuation of the study drug. There were 14 serious adverse events reported. 1 was related to study drug and resulted in discontinuation. The ones that the adverse events that resulted in premature discontinuation of the drug included peripheral edema, hyperglycemia, hirsutism, fluid retention edema associated with pneumonia. When we look at the most common reported adverse events, we found that these included hypertrichosis, peripheral edema, hyperglycemia, hirsutism, increased blood glucose, and headache. When we specifically look at hypertrichosis, peripheral edema, and hyperglycemia, you can see that they were all grade 1 and grade 2 and did not reach severe levels. When we look a little bit closer to fasting glucose, we do see that there was an initial rise in fasting glucose from baseline up to 13 and 26 weeks, but that over time this did improve, and by month 15 it was similar to what it was at baseline. This similar pattern was also seen for hemoglobin A1C. To summarize what we found that DCCR improves hyperphagia, as well as improves several other PWS-related behaviors and cardiometabolic parameters in subjects with PWS. These improvements contain and sustain over 1 year. And when we compare these results to results from PATH for PWS as a control, a natural history control, we show that there is clear divergence. And we found that DCCR is generally well tolerated with an expected adverse event profile. And overall, we found that these results were quite promising. These are the sites that were involved in Destiny PWS. I would like to thank all the subjects and their caregivers who participated in both Destiny PWS and PATH for PWS. I would like to thank all of the investigators and study personnel for Destiny PWS and PATH for PWS. And we are now open for questions. Thank you very much.
Great, thank you so much, Parisa. So we do have a couple of questions. As a reminder, if you have any questions, please put them in the chat and I'll ask the presenters. So the first one, Parisa, is did any patient develop diabetes or suspected diabetes with a hemoglobin A1C greater than 7% during the DCCR study requiring decrease of dosing?
There was a handful of patients who had elevated A1C, but as you can see, it did come down. There was also a handful of patients that had to start metformin to help with glycemic control. But overall, after a period of time, they did well.
Okay, great. And Virginia Camonis asks, can you share the AEs and SAEs found in the placebo group?
I don't have the slide in front of me, but they were very similar in terms of certain things, except for the ones that we pointed out that more than 5% of subjects had. So in terms of the hypertrichosis, hyperglycemia, and the peripheral edema. I don't have this slide to give you more details on the placebo group.
Okay, great, thank you. You know what, one of the things that was so impressive to me is the durability of this response. And I believe your site was one of the first sites. So, so maybe you can just comment on that a little bit more. Do you see any of the participants kind of bouncing back, or are they staying pretty stable?
Actually, one of the really remarkable things is that they continue to do better over time, our subjects. So I find that this is a drug, I like to call it a marathon, not a sprint in terms of effects. So the more time they're on the drug, it seems to be that they have more of the beneficial effects and more long-lasting effects. So and I do have the subject in our study that's been on it the longest and continues to do well with no bounce or rebound hyperphagia.
Great, and we're running out of time, but I'll just ask one more from Jan Forster. What are your thoughts about the robust improvement in mood and behavior? And well, let's just keep it at that. And then there's a couple more questions in the chat that maybe you can respond to after. But what are your thoughts about why mood and behavior improves?
Well, Dr. Forster is more of an expert in this than I am, of course. But I do, you know, just as a clinician, when I see my subjects are having less issues with hyperphagia, I feel like that helps with a lot of their behavioral problems. Less focus on food and less focus on being hungry all the time helps, seems like, to decrease some of the other behavioral symptoms. That's my best guess as an endocrinologist. So I do feel like a lot of the irregular behaviors, atypical behaviors we see in PWS could be linked to their just constant hunger, and being able to alleviate some of those symptoms helps overall.
I think that's one of the big mysteries, is how much is due to hyperphagia and how much is separate. And maybe with some of these new drugs, we'll finally be able to tease that apart a little bit. So thank you very much for your comments. And again, if you there's a couple more questions in the chat, if you get a chance to do that, we definitely appreciate it.
Sure.
Okay, so we'll move on now. Our next speaker is Rachel Weverick from University of Alberta, and she is going to talk about the potential role of the N-terminal domain of the Shaft-Yang syndrome protein MAGL2 in RNA metabolism.
Thank you all for coming, and thank you to FPWR for giving me this opportunity to present our study.
Welcome, everybody. I'm delighted to present at the FPWR scientific meeting this year. And today in my presentation, I would like to talk about the very intricate relationship between Shaft-Yang syndrome and Prader-Willi syndrome and what we can learn from each other. It's very unfortunate that, again, we cannot meet in person, but I'm sending my very warm regards from beautiful Heidelberg in Germany. And hopefully next year we can be in a post-COVID world and meet in person again for some scientific exchange and also personal exchange. So the story that I'm telling today all goes back to 2013, when we first reported in Nature Genetics 4 individuals who had a clinical phenotype that reminded us of Prader-Willi syndrome, but were found to have very unusual mutations, being point mutations, truncating point mutations of the MAGEL2 gene. MAGEL2 is one of the protein coding genes within the Prader-Willi syndrome domain on chromosome 15q, and MAGEL2 is maternally imprinted, paternally expressed, as is typical for the Prader-Willi locus. At the time, we started to characterize the clinical phenotype of those individuals, and we felt that there was overlap between this condition caused by point mutations in MAGEL2 and classic Prader-Willi syndrome, the overlap being hypotonia, feeding difficulties, developmental delay, intellectual disability, and hypogonadism. We felt that in our initial cohort, there were also classic Prader-Willi phenotypes missing, in particular hyperphagia and the morbid obesity if going untreated. On the other hand, in the individuals with the MAGEL2 point mutations, later called Schaaf-Yang syndrome, we did see a much higher prevalence of autism spectrum disorder, and we did see contractures, especially affecting the small finger joints. Since 2013, we have gotten in touch with many more individuals with MAGEL2 mutations. And in our database at this point, there are more than 250 individuals with truncating mutations of the MAGEL2 gene. The last time we reported a cohort of, I think at that time, around 120 individuals was published in the American Journal of Medical Genetics by one of my students, John McCarthy, and team in 2018. What is unique about Schaaf-Yang syndrome in comparison to Prader-Willi syndrome is that we do see familial cases because obviously this MAGEL2 truncating mutation can run in the family as long as it is inherited on the maternal line. But when a father passes on the truncating mutation of MAGEL2, then those children will be clinically affected. So we're in touch with multiple families who have multiple affected individuals, including some large families as the European family you see on the bottom here. What are the clinical features of Schaaf-Yang syndrome based on our understanding in 2021? Well, in infancy and childhood, it looks very much like Prader-Willi syndrome. There's low muscle tone and feeding difficulties. Many boys have undescended testicles, and as the children age, we do see developmental delay, intellectual disability, and a relatively high prevalence of autism spectrum disorder. These are also some of the differences compared to Prader-Willi. The prevalence of autism spectrum disorder in Schaaf-Yang seems to be much higher, around 70 to 80%. And in addition to the typical Prader-Willi features, we have the contractures, in particular those of the hands. Additional common features of Schaaf-Yang syndrome include sleep apnea, which can be obstructive, central, or combined, and is present in about 3/4 of all individuals. We have a very high prevalence of feeding problems, almost affecting all of those children, high prevalence of dysphagia, and around 3/4 of children go through some time of requiring tube feeding. Respiratory problems are very prevalent in Schaaf-Yang syndrome. I think they are more prevalent in Schaaf-Yang than in Prader-Willi. Almost 60% of those children have a history of intubation, and almost 1 in 5 children has a history of tracheostomy. These are the hands of Schaaf-Yang syndrome. They can be fairly severe, clenched hands. Some of them have required surgery to release the tendons and allow for an opposing thumb. But sometimes they can also be mild. But you see the typical tapering fingers and the typical contractures of the small finger joints here. Other skeletal issues include a decreased bone mineral density, and we do have a high prevalence of scoliosis. Many of the Xiaofiang children have required surgery to correct the severity of the scoliosis. Now, only in the last few years we have come to learn about the adult phenotype of Xiaofiang syndrome. This is in the nature of the situation because exome sequencing is mostly applied to children rather than adults, sometimes not covered by insurance companies for adults. So our initial cohorts were very heavy on the pediatric side, and only over the years we've come into touch with more and more adult individuals affected by Xiaofiang syndrome. This paper was published last year in the Orphanet Journal, and first author is Felix Marbach, who's a young physician working in my team here in Heidelberg. Looking at these young adults and kind of mid-age adults, you can see that the phenotype of Xiaofiang syndrome in part changes over time. Medically, I should say, Xiaofiang syndrome becomes less complicated over time, but then there are additional phenotypes that seem to arise over time. In particular, the obesity. So a lot of the adult individuals with Xiaofiang syndrome are overweight. And in addition to that, or as being the cause of the obesity, we do see a fairly high prevalence of overeating and food-seeking behavior. Other phenotypes that persist include the sleep apnea and the respiratory difficulties, also the abnormal sleep cycle, changes in the circadian rhythm, etc. Also, as individuals with Xiaofiang syndrome get older, there appear additional behavioral phenotypes, and it seems that a lot of these adults with Xiaofiang syndrome do have a relatively high prevalence of psychiatric phenotypes, including sometimes aggressive behaviors, temper tantrums, stubbornness, a lot of overlap there with Prader-Willi syndrome. Also manic episodes, depressive mood, a high level of anxiety, social withdrawal, restricted interests, etc. So to some degree, our perspective on the clinical phenotype of Xiaofiang syndrome has changed over time. This was our initial perspective of 2013 until fairly recently. But I would argue that there aren't that many phenotypes of Prader-Willi syndrome left that we do not see in Xiaofiang syndrome. In fact, the more that I learn about these individuals, the greater the overlap appears to be. So I think clinically I've been able to show you that there's an intricate relationship between Prader-Willi syndrome and Xiaofiang syndrome. How can we use that knowledge to also consider therapeutic avenues that may be shared between the two? So we do know from individuals with Prader-Willi syndrome that growth hormone deficiency is very common, and growth hormone therapy with recombinant growth hormone is one of the approved therapies and mainstays actually of therapy for Prader-Willi syndrome. In a cohort of only 10 individuals that we reported in 2018, investigating IGF-1 and IGF-BP3 levels in individuals with Xiaofiang syndrome, there was the indication that yes, indeed, it seems that a lot of kids with Xiaofiang syndrome do have growth hormone deficiency. So as mentioned, growth hormone is an FDA-approved treatment for Prader-Willi syndrome. So we wanted to learn about the individuals with Xiaofiang syndrome and their experience with recombinant growth hormone, as more and more of these individuals have been put on recombinant growth hormone as an off-label use. Initially, this was all based on individual case reports, and we see one of the girls with Xiaofiang syndrome here, Trinity, and there's only a few months of time between these two pictures after she'd been started in growth hormone, and you can see what tremendous effect it has on her body composition, stature, but also the ability to perform certain motor tasks and kind of, have the endurance and stamina to walk long distances, etc. So we felt that there was a need for a more systematic approach, and we performed a retrospective analysis of growth hormone therapy in children with Schaaf-Yang syndrome, which was published earlier this year by one of my medical students, Niels Hebach, and our team. We obtained information from a total of 26 individuals with Schaaf-Yang syndrome, 14 of which had been on recombinant growth hormone, and 12 individuals were without treatment. And you can see here in these box plots on the right that treatment with recombinant growth hormone has a significant effect on longitudinal growth, as looked at by height z-score, and also the change in height z-score over time. You can see that only 3 and 6 months after the treatment start, there's already a significant difference between before and after. And also there's a significant difference in height z-score between the untreated and the treated group. Recombinant growth hormone also has a positive effect on body mass in individuals with Schaaf-Yang syndrome. And you can see on the right side here, time after treatment start, 3 months and 6 months, there are significant changes in BMI z-score after recombinant growth hormone therapy has been started. More than that, recombinant growth hormone has a positive effect on strength and stamina as reported by these families. And if you look at the bottom graph here, you can see that the satisfaction of recombinant growth hormone therapy in Schaaf-Yang syndrome is fairly high. There are few individuals who report that to be neutral, but most of them are satisfied or even very satisfied with the treatment. So that's what we've learned from Prader-Willi in regards of Schaaf-Yang, but what can we learn from Schaaf-Yang research, possibly translating to Prader-Willi syndrome as well? So based on mouse models and also induced neurons generated from individuals with Schaaf-Yang, we found that there's an upregulation in the mTOR pathway. And because mTOR can be downregulated by drugs like rapamycin, we felt that it would be worth treating those neurons with rapamycin to see if we can rescue the effect and kind of bring it back to a normal level. And yes, indeed, that is the case, as published by my PhD student, Emmeline Crutcher, in 2019. Rapamycin treatment rescues the morphological phenotypes in regards of dendritic branching and intersections of those neurons that we see in individuals with Schaaf-Yang syndrome. Based on those initial findings, we are currently studying induced neurons from individuals with Prader-Willi syndrome and neurons that have been generated to have Prader-Willi deletions. But also we are performing a study that is funded by FPWR, where we perform an in vivo treatment prenatal in one cohort and postnatal in the other cohort of an Schaaf-Yang syndrome mouse model, the MAGEL2 knockout mouse, the one generated by Rachel Bebrick and Stuart Collins and team. And we want to find out in the postnatal treatment arm if we can rescue muscle strength and endurance. So this study is still ongoing, but I want to show you our treatment paradigm and initial findings. Treatment with rapamycin is starting on postnatal day 10, and then we do IP injections with rapamycin, a lower dose for the first 2 weeks or so, and then we go up to a higher dose and treat 3 times a week. And at around 14 weeks of age of those mice, we do a test of motor function. And our initial experiments show this. So this is treadmill performance of those mice, and you see in purple the wild type treated with placebo, in light blue the MAGEL2 knockout mouse treated with placebo, in red the wild type mouse treated with rapamycin, and in yellow the knockout mouse treated with rapamycin. And what you do see is that there's a significant effect, and that it appears that at least there's a partial rescue of the phenotype that we see in treadmill performance for the MAGEL2 knockout mouse that we can overcome with rapamycin treatment, which I think is very promising. And we're continuing those studies and also looking at other motor phenotypes, and hopefully we can report that back to you soon. So in summary, I showed you that Prader-Willi syndrome and Schaaf-Yang are molecularly and phenotypically related. The Schaaf-Yang syndrome phenotype may be more similar to Prader-Willi than initially thought. I showed you that recombinant growth hormone proves beneficial in both entities, and I showed you initial findings from our investigations of mTOR upregulation, which are still ongoing. With that, I will close and acknowledge my teams, both in Houston and in Heidelberg. I would like to thank the Foundation for Prader-Willi Research for their generous funding, and of course, the wonderful families we're in touch with who provide the inspiration to everything that we do. And with that, I'll close and look forward to the live Q&A.
Great. Thank you, Christian, for that presentation. We have a few questions coming in. Anthony Isles asks, do children with SYS develop obesity at higher rates than individuals with learning disabilities in general?
I do think so, but we have not looked at that systematically. In particular, I would highlight again that the obesity in Schaaf-Yang syndrome is of a relatively later onset. It seems that they really start crossing percentiles in their teenage years and young adult years.
Do you have any thoughts about why that is? Is that, do you have any thoughts?
Yeah, I don't know. We've asked that ourselves. One hypothesis that I have is that Schaaf-Yang kids seem to be cognitively more impaired than Prader-Willi kids, and especially during younger childhood, early childhood, they simply don't seem capable of food seeking. They're so much kind of limited to what they are given by their parents that they have no chance to really develop
A question about, is there evidence of oxytocin alterations in Schaaf-Yang?
There's evidence of oxytocin alterations in the MAGEL2 knockout mice, as has been studied by several groups, but we have not done any assessments of oxytocin in human individuals with Schaaf-Yang syndrome to date.
Okay, great. And Larry Ryder asks about the ASD incidence. We know it's higher in Schaaf-Yang, but do you think that's driven by the OCD behaviors or other domains of ASD? We do know that, in those kids who have been tested, that it's not just autistic features, but they do actually meet criteria based on ADAS and ADIR. I'm not sure it's hitting any particular domains. It really, to me, seems to be kind of almost like a garden variety autism spectrum disorder. Obsessive compulsive behaviors. Maybe I'm not an expert enough to really, say whether those are more prevalent in Schaaf-Yang syndrome than, what you would see in kind of your average child with ASD. I do see a ton of kids with ASD in my genetics clinic, and regarding the profile of behaviors, I don't see much difference between Schaaf-Yang and the others, actually. Okay, great. Maybe one more question from Diane Stafford. She asked, there is efficacy using growth hormone in Schaaf-Yang kids, but have you looked for evidence of true growth hormone deficiency?
So, looking at these 24 children, we obviously looked at how were they assessed before they were given recombinant growth hormone. Most of them did get IGF-1 levels tested. Only 25% actually had a stimulation test being performed and were shown to have, formal growth hormone deficiency. And there's there was one kid in the group who did not have any endocrine testing at all, and just based on the short stature was started on recombinant growth hormone.
Okay, great. Thank you. I think we'll go ahead and move on, but if you get a chance, Christian, to check the chat, there's a couple more questions that you might want to address.
Yes, thank you.
So apologies, because we do seem to have a little bit of in and out with the sound. So please bear with us as we try to get all of that fixed. And we'll go back to Rachel Webrook now from the University of Alberta, talking about the N-terminal domain of MAGEL2 in RNA metabolism.
Thank you all for coming, and thank you to FPWR for giving me this opportunity to present our study. This study was performed by Matea Sanderson, who was a PhD student in my research group at the University of Alberta in Edmonton, Canada. Matea had assistance from Jocelyn Bischoff and Richard Fallman, who are also both at the University of Alberta. We gratefully acknowledge funding from the Foundation for Prader-Willi Research for this study. So as you all know, Prader-Willi syndrome and Schaaf-Yang syndrome have overlapping phenotypes that include neonatal hypotonia with failure to thrive, developmental disabilities, and disruptions to hormone regulation. Each of these two disorders also has unique clinical findings, and they are both considered rare disorders, with Schaaf-Yang syndrome being an ultra-rare disorder. Along with the overlapping clinical findings, there are also overlapping genetic causes. So Prader-Willi syndrome is caused by disruptions to the paternal copy of 15q11 to q13, usually a microdeletion that includes the MAGEL2, nectin, SNORD116, and other genes. In contrast, Schaaf-Yang syndrome is caused by loss of function or mutations in the MAGEL2 gene alone, which is in the MAGEL2 gene being inside the Prader-Willi syndrome deletion region. MAGEL2 is a member of a family of about 40 proteins that share a MAGE homology domain, which is a protein-protein interaction domain. The nectin protein is also a member of this same family of proteins. And as I mentioned, MAGEL2 is mutated in Schaaf-Yang syndrome, while nectin and MAGEL2 are both deleted in Prader-Willi syndrome. Some MAGE proteins have redundant functions, and mutations in other MAGE proteins are involved in genetic disorders, while still other MAGE proteins are mutated or dysregulated in cancer. We know that MAGE proteins serve as adapters for ubiquitination-mediated processes. MAGE proteins bind to E3 ubiquitin ligases and bind to D-ubiquitinases to modify the ubiquitination, seen here with these orange balls, of substrate proteins. The L2 MAGE protein, specifically the MAGEL2 protein, regulates the ubiquitination of substrates important for actin dynamics, endosomal trafficking, circadian rhythm, and many other processes, while other MAGE proteins are important for the processes that are shown here. We know that MAGEL2 is expressed in many different structures in the brain. It's primarily expressed in the hypothalamus, which is important for appetite, regulates circadian rhythm, regulates hormone production. And so it's perhaps not surprising that these are the processes that are mainly disrupted in Prader-Willi syndrome and in Schaaf-Yang syndrome. MAGEL2 is also expressed in developing muscle and cartilage and bone. So as I mentioned, mutations in humans cause Schaaf-Yang syndrome. When mutated in mice, we see increased fat mass, decreased muscle mass, and abnormal behavior, as well as hormone dysregulation. The MAGE homology domain of MAGEL2 has been extensively investigated in cellular assays by itself. These assays use a truncated version of MAGEL2 that contains only this C-terminal half of the protein. So the question for our study is what does the rest of the MAGE-L2 protein, and in particular, what does the N-terminal region of MAGE-L2 do? As I mentioned, most Schaaf-Yang syndrome mutations truncate the MAGE-L2 protein, and so they delete the MAGE domain with leaving the N-terminal domain intact. MAGE-L2 is completely inactivated in Prader-Willi syndrome. The MAGE domain here is shown in green, and as well there's a USP7 binding region shown in purple, and that is part of the ubiquitination process. We looked first at the N-terminal half of MAGE-L2, first at amino acid composition. And what you see here is that there is an enrichment in these proline-glycine dinucleotides and proline-XN glycine motifs, so the red stars and the black stars at the top of the diagram. And these are marks of an intrinsically disordered proteins. These types of proteins are prone to phase separation, and in fact, they have high P-scores indicated in the middle chart, where you see the predicted phase separation propensity or P-score. These types of proteins are contained in membraneless organelles that are supramolecular assemblies of proteins and RNA molecules. The bioinformatic tools that we used also show a high disorder probability in the N-terminus, and that's shown on the bottom, and that also is a function of the amino acid composition of the N-terminus of the protein. So as we were performing this analysis, we were motivated by this report in Cell, where these researchers stated that intrinsically disordered protein regions in proteins are prone to phase separation, and that mutations in these may account for missing heritability, specifically in autism spectrum disorders. Mutations in intrinsically disordered regions change protein partitioning within the cell. They impact biological function, including many functions that are relevant to Schaaf-Yang syndrome and Prader-Willi syndrome, including activity-dependent translation and synapse organization and other processes in the brain. And you can imagine how mutations that separate the structured domain of MAGE-L2 from the intrinsically disordered regions could cause disruptions to these subcellular phase-separated compartments. So proteins are not present in isolation in the cell, but rather they are surrounded by a cloud of other proteins and RNAs. We and others have found proteins that are present near the C-terminal region of the MAGE-L2 protein, but we hadn't previously identified any that are near the N-terminus of this protein. To further investigate this, we used a method called BioID, which is a proximity labeling and mass spectrometry method to identify these proteins in cultured cells. So in this experiment, cells are stably transfected with a tetracycline-inducible form of MAGE-L2 fused to biotin ligase. The cells are then cultured with an excess of biotin, and the biotinylated proteins are recovered using a streptavidin bead-based process processed by mass spectrometry to identify the proteins that pass within about 10 nanometers of MAGE-L2. After performing multiple replicate experiments and processing through the data using the software, proteins are broadly classified into biological processes that include RNA processing, ubiquitination, protein transport, and cell signaling. It can be very challenging to determine whether a missense mutation that changes a single amino acid is in fact pathogenic. To address this question, we constructed a MAGE-L2 protein that has a single amino acid change that recapitulates a disease-causing mutation in another MAGE protein. The location of this protein, which is an arginine to cysteine at 1187, is shown with the red arrow. The network of proximal proteins was altered by this mutation, demonstrating that BioID and other protein-based approaches can be used to categorize variants of unknown significance that are found in MAGEL2 in children with neurodevelopmental disorders or autism. As mentioned previously, most studies to date have examined a C-terminal fragment of MAGEL2 containing the MAGE homology domain. Here we did an experiment where we looked at the full MAGEL2 protein and then subtracted proteins that were present in the bioID assays from the C-terminus of MAGEL2. So you can see that there are proteins that overlap between these two prey proteins, and then there are proteins that are unique to the full-length protein. Two clusters of protein caught our attention and are highlighted here in yellow. The YTHDF proteins promote stress granule formation by binding to a modified N6 modified methyl adenosine on RNAs. Stress granules are aggregates composed of proteins and RNA molecules, including stalled translation initiation complexes. Stress granules form in cells as part of normal cellular responses to stress. The TNRC6 proteins, which are down on the right, function in post-transcriptional gene silencing through the RNA interference and microRNA pathways. These proteins bind messenger RNAs in cellular compartments called P-bodies. The N-terminal region of MAGEL2 associates with all three of the YTHDF proteins and both of the TNRC6 proteins. This is consistent with the bioinformatic prediction that the N-terminal region of MAGEL2 is in phase-separated compartments, membraneless compartments inside the cell. We next performed a series of cell biological and biochemical experiments to demonstrate the physical and functional associations between MAGEL2 and key RNA binding proteins. Two of these experiments are shown here. But first on the left is a representation of a cell with the nuclear and cytosolic compartments labeled, and you can see that the YTHDF proteins are in the cytosolic compartment where they facilitate translation and mRNA decay. These YTHDF proteins shift to the nucleus under conditions of cellular stress. So on the top right are the results of an experiment where we applied a heat shock stress to cells and then followed the distribution of YTHDF2 protein between the cytoplasm and the nucleus by cellular fractionation. After heat shock, the bulk of the YTHDF2 protein moves to the nucleus and then slowly returns to the cytoplasm, as shown by the black curve. However, when MAGEL2 is overexpressed, the intracellular distribution of YTHDF2 protein after heat shock stress is inhibited, shown in the red curve. In the bottom right panel, our results show that fibroblasts that are derived from individuals with Prader-Willi syndrome who don't have any MAGEL2 protein have less YTHDF2 protein than fibroblasts from control individuals. This suggests that MAGEL2 has roles in stabilizing the YTHDF2 protein and also regulates cellular responses to stress through modifying the intracellular distribution of the YTHDF proteins. In summary, MAGEL2 is deleted in Prader-Willi syndrome and mutated in Schaaf-Yang syndrome. The C-terminus of MAGEL2 functions in endosomal trafficking and in ubiquitination, while the N-terminus of MAGEL2 contains an intrinsically disordered protein domain that is predicted to undergo phase separation into membraneless compartments. We also know that disruptions to intrinsically disordered proteins contribute to autism spectrum disorder. MAGEL2 interacts with and regulates YTHDF and TNRC proteins, which modulate stress responses through mRNA binding and nucleocytoplasmic transport. So this study by Matthias Anderson was just published in the Journal of Biological Chemistry. Anybody who is interested in more details than this short talk can provide can see all of the data in that publication. Thank you for your attention, and I would be happy to answer any questions that you might have. Thanks.
Thank you, Rachel. I'm actually going to jump back to a question that Rob Nichols had for Christian, because I think it's relevant to your talk as well, about are the more severe phenotypes in Schaff-Yang syndrome perhaps due to the toxic effects of a truncated MAGL2? Did you want to comment on that one?
Sure. Yeah, that's definitely a possibility, because the N-terminus of the protein is likely to still be translated, while the C-terminus of the protein is no longer there. It's a single exon gene, so it's less likely to be subject to RNA-mediated decay of the RNA. So it's quite possible that that region is indeed translated. And what we need to do is develop some more antibodies, actually, to look at that question more precisely. But it's quite possible that there is a toxic effect of separating the N-terminus from the C-terminus in that protein.
Great. And Larry Ryder asks, what cell type did you use for BioID? And also, why do you think the mutation caused the loss of the USP7 interaction?
Sure. So the cell type was a specialized derivative of HEK293 cells, and that's because it's a single copy integration into a fixed site in those cells, and there are not very many available cell types around that where you can actually do that, do the experiment the way we wanted to do it, with single copy integration and with the same site always of integration. I think going forward, we do need to look at many other cell types to really get an idea of what the protein-protein interactions are like in more relevant cell types than the ones that we used. And that's definitely something that needs to be done going forward. And then the second question was
about the USP7.
So the mutation that particular one that I showed is in the C-terminus of the protein, and so affects the structure of the MAGE homology domain. How it affects the USP7 binding domain could be just because it's an adjacent domain, and so it probably destabilizes the structure of that whole area. We actually published a paper on nectin using an analogous mutation and looked specifically at the structure, and that was in just published a year ago. So if you actually want to look at the structure of the MAGE homology domain with that particular mutation, then that data is actually in the paper on nectin and how mutations affect protein-protein interactions.
Great. And Marnie Blewett asks, have you used Ryan Potts's MAGL2 antibody to look at staining in the nucleus? And this is related to the issues around the phase separation.
Yeah, and that's definitely something that's ongoing and something that we need to look at. We can't actually look at the endogenous protein because the HEK293 cells don't have endogenous MAGL2. So we'd be looking at it in the context of a heterologous system. But I mean, that experiment would probably best be done in cells from directly in cells with individuals with Schaff-Yang syndrome. Yeah.
Great. And Rachel Gilmore asks, do the plasmids with MAGL2 include the entire MAGL2 transcript, introns? Well, there are no introns, 3', 5' UTRs, or just the coding sequence?
So there are no introns, and it's either the whole coding region or just the C-terminal half of the protein, but no untranslated regions in those. It's really a protein interaction assay. It's not really designed to look at any of the rest of the transcript.
Great, and maybe we'll take one more, and then there's a couple left in the chat box if you want to answer there. But does MAGL2 protect viability of cells under stress?
Yeah, that's a really good question. I mean, the flip side to that question is, does the mutant protein, the short protein, actually affect the viability of the cells? So does the wild type protein improve viability, and does that truncated protein affect viability as well. And that's not something we've looked at directly, although we do know that the cells that we have that express the mutant protein survive and are not notably different in their doubling time or anything. And there are mice and people alive with this mutation. So I don't think it has a severe effect on cell viability, or else it wouldn't be viable at the cellular or at the animal level.
Great. Well, thank you. Well, we'll let you, there's a couple more in the chat box, and we'll let you answer those sort of offline, because we are wanting to get to a bit of a break. I have just a little bit of housekeeping before the break. I did want to mention, and I think we have a couple of slides, the back of your abstracts book has some links to some research resources that you may find useful, everything from molecular resources. I'll note that we have a partnership with Autism BrainNet, and my understanding is there are now 9 different brain samples that are available through that resource. So obviously this is a very precious gift, and we appreciate the families who chose to donate those brains, and we'd love to see that tissue used to advance our understanding of PWS. There's also on our website cell lines and animal models. It may not be complete. If there are other cell lines and animal models that you have that you'd like us to add, we would love to do that. And then another resource from the Writer Lab is, I think, on the next slide, is a biomaterials from dental pulp stem cells that the Writer Lab has developed. So next slide. So we provided a bit of funding so that Larry's group could take some of the dental pulp stem cells that they've developed. So they have a lot of lines, deletion, UPD, with and without autism. They can differentiate those cells. They've got RNA and protein, and then they're able to share that with the research community. So if you want to, double check some findings and use a different source of patient-derived cells, they may be a really nice resource. I think the contact information is on the last slide for the Writer Lab. We also have on the FPWR website, we have a little brochure you can download and look at those different cell lines that are available. So again, just some resources that may be helpful to you. If there are other resources that you think of that would be good for the entire PWS community to share, we'd be interested in hearing your thoughts on that. So we'll take about a 10-minute break now. When you come back, there are concurrent sessions, and you can choose to either be in the molecular track, which I believe is on this landing page, or if you scroll down to the bottom, there's a button for the clinical track, and we'll have a few more presentations. Again, concurrently, you can bounce back and forth between the sessions, and also all of these presentations will be made available after the conference. So if you miss anything, you'll be able to go back and listen again or catch anything that you missed. So we'll take a 10-minute break, and then please choose molecular or clinical. We thank all of our speakers so far. It's been and thank you all for the questions. It's been as lively as you can get in a virtual session. So we really appreciate that. And we'll see you in about 10 minutes.
Hi, everyone, and welcome back. This is the genetics and molecular session. If you are in the wrong place and would like to be in the clinical session, please head to the bottom of the landing page in order to get to that session. So first, I'd like to turn it over to our first presenter for this genetics and molecular talk. We have Siptane Haider from the University Medical Center of Freiburg, and he's going to be talking about precise epigenome editing as a novel therapeutic approach for Prader-Willi syndrome.
Hi everyone, I'm Siptane Haider. I'm doing my PhD in Institute of Transfusion Medicine, Medical Center, University of Freiburg, and today I'm going to present a part of my project, which is precise epigenome editing as a novel therapeutic opportunity for Prader-Willi syndrome. First, I will give you an overview of Prader-Willi syndrome. Very simple, that Prader-Willi syndrome is an imprinting disorder caused by loss of function of specific gene in chromosome 15. It can be happen because of deletion of parental allele or uniparental disomy, or maybe imprinting of parental allele. But in our project, we are more focused on this maternal allele, which have all the copies of these genes but silenced because of imprinting. So we will use molecular techniques or epigenome editing to reactivate this maternal allele. Just like some drugs-based therapy that are available, in which they use methylation inhibitors or DNA demethylase, histone demethylase, methylation inhibitors to reactivate maternal allele. And although they are working and there is expression of SNRPN gene in maternal allele or some other locus, but there is a big drawback in their approach is that genome-wide off-targets and epigenetic toxicity, which is not favorable. In our approach, we suggest using specific epigenome editor that will target only Prader-Willi IC region or Prader-Willi critical region. This will lead to not only very specific local effect and also nearly no side effects or no off targets. So first of all, I will just go an overview of my project outline. In my project, the first step is the identification of best epigenome editor. For that, we have a reporter system in our lab. This reporter system has methylation of PDCD1 region, cis element, upstream to tag BFP. Because of this methylation, this tag BFP is silenced through epigenetically. Now, I will use a tail specific for PDCD1 and combine it with different activators. These activators can be DNA demethylase, histone acetylase, or histone demethylase, or maybe a fusion of them. So I will combine them and try to reactivate this tag BFP. In this way, I will end up a fusion or some effector that can actually activate this epigenetically silenced tag BFP. In next step, we will design multiple tails that will target this Prader-Willi region. The most tail we will design on this Prader-Willi IC region, SNRPN, and SNOD116 region. In third step, we will combine these tails and the activator from the first step. In this way, we will have specific activator for Prader-Willi critical region. These activator then will be transfected into fibroblast, and their effect on these SNOD116 or SNRPN will be checked using qPCR and ddPCR. Out of these tails, those tails who are working best or most efficient can be taken into patient-derived IPCs, and their effect will be studied in neuron cells and all that. This was an overview of my project. Now, to identify different epigenome activators, I use 3 different classes: DNA modifiers, transcription activators, and histone modifier. I make single fusions, double fusions, and also sometimes triple fusions, and try to reactivate the reporter system. Before taking them into the reporter system, we also confirm their expression, their size, if everything is good, through Western blotting, and when they're ready, we take them for transfection in reporter cell. This is just a general outline for our transfection. We use pi transfection and some pi transfection in our reporter system, and all the activators we prepared, we take them into reporter system and did Fox analysis at different time points. To analyze Fox data, there are different and this is a simple template in which you can see that these are wild type hex cells and our positive cells, and this gate for untreated and treated cells after treatment of our activators. And this simple graph represents the MFI of all living cells and different fusion that we use in the cell. We use this MFI to compare best working effector or the combination. This was the initial data we got through transfection, and it helped us to narrow down some 4 or 5 major fusions that we did further analyze using lipofaction. In lipofaction, as the transfection efficiency increased, also the expression of tag BFP also increased, and it helped us to pinpoint the most working effectors, and we get our 3 best effectors which were able to reactivate our reporter systems. Next thing, we also wanted to see if these fusion can also activate some locus, endogenous locus, as our tails are targeting PDCD1, which is only expressed in hex cell. So we thought, like, why not check their expression in hex cell in our treated cell? If our tails are binding to this PDCD1 locus, they can activate the PDCD1. And we did RNA extraction at day 5 and qPCR analysis. Surprisingly, qPCR, their expression of PDCD1 was really high, which show that how much our effectors can upregulate the expression of indigenous locus. And our fusion not only can activate synthetic promoter, but also endogenous locus. Last thing I want to focus here is that all these results are from plasmid, not with mRNA. With mRNA, the efficiency will increase much more, and we have some experiment in pipeline, but in the end, we have our best effector that are working in reporter system and also for endogenous locus. Next step was to take them into more reliable system like primary fibroblast. So now we come to fibroblast, which express SNRPins, Node116, and UBE3A. All these genes, they are very important in Prader-Willi case, that's their expression, but we unfortunately don't have patient-derived fibroblast. Thanks to our collaborator, we did receive an RNA from a patient. This patient has a small SNOD116 deletion. So we use this patient-derived fibroblast RNA as to establish qPCR in our lab and compare it with wild-type fibroblast. This is just an overview that how the qPCR results will look like. It shows that cDNA on fibroblast, in patient-derived fibroblast and control samples, how much SNOD116 expression is less as compared to control, but the expression of SNRPin was not so much disturbed. Actually, it was same. And it also shows that our primer was specific and we can use NUF-based system to compare the expression of SNOD116 and SNRPin. While we are waiting for our patient-derived fibroblast, we also decided to design tail on this region. We designed nearly 20 tails on this SNRPin, SNOD116, and Prader-Willi IC region. And then we thought, why not we take them into wild-type fibroblast and check some of our tails are binding or not. So to confirm the binding of our tails to this region, we not only combine this tail with our activator, but also with DNA demethylase. This leads to a double verification. With DNA methylase and grab the expression of gene downregulate, and with our activator, the expression of that gene should activate. And we transfected these both combinations into wild-type fibroblast. And after 5 days, we checked the expression using qPCR. This qPCR analysis of our transfection shows that in control, as in SNOD116, the expression was not so much change, and our tails, the tails we used, did not affect the SNOD116 expression. But when we see the expression in SNRPin, it follows the trend we were expecting. The GRAB domains and DNMT3L cause the downregulation of SNRPin gene, and our reporter increase the expression of SNRPN gene. So this shows that our fusions are also working very good in fibroblast, in primary cells, and now we are ready for patient-derived fibroblasts, which we are expecting from our collaborators soon. And then we will take our fusions and our DNA binding domain of fibroblast specific into patient-derived fibroblast. In last step, I will just summarize my talk that our fusion that we made through this reporter system are not only able to activate the synthetic reporter system, but also endogenous locus. And our initial result on wild-type fibroblasts show that our effectors are working efficiently in primary cells. We have nearly 20 tails for targeting SNOD116, SNRPN gene, and also some regions which are repetitive in this region. We are waiting for our patient-derived fibroblast with large deletions, so we can actually test all these tails. We are also developing a DCAS9 based platform. This platform will be much more easy to screen all these regions and may help us to pinpoint the specific regions controlling this Prader-Willi downstream genes, or can be Prader-Willi, how they are important in Prader-Willi syndrome. So that's why this approach is not only a therapeutic opportunity, but can also lead to identification of some new regions or important imprinted regions. In last, I would like to thank my supervisor, Dr. Claudio Mussolino, who gave me an opportunity to work in this exciting project. My labs senior, Silvia Romana, she helped me a lot. Prader-Willi Foundation, who gave funding for this project, DAAD, who gave me funding to come here and work, my institute, ITG, University of Freiburg, and all of you for listening my talk. Now we are ready for some Q&As if you want. Thank you.
Thanks so much, Subtain. Just as a reminder, if anyone has any questions for any of our presenters, please put them in the chat. So I guess one question that I had is related to the types of effectors. Which one do you think would either work best, or which one do you think which type is easiest in order to move into a transition into more of like a human or clinic aspect?
Yeah, for effectors, I think DNA demethylase and histone acetylase, they work best because to stop DNA demethylation coming back, we used a P300, which is an acetylase. So it acetylase those histone tails and stop them from remethylation. And also for DNA, it's also demethylation was our very constant. Like we see keep them for 22 days, and there was no coming back of expression. So I think in this TET1 and P300, they were really good. But one thing is VPR. If you know, it's a combination of VP64 and two other effectors. It was much more effective if you want a robust or activation. For example, they were activated. Activation was very fast in 2 days. With TET1, it takes like 8 days for reactivation of our reporter system. But with VPR in a fusion, so the activation was in 2 days. We can see the expression and change all that.
Oh, interesting. Okay, great. Stormy Chamberlain asks, do you think altering DNA methylation will be possible in post-mitotic cells?
Yeah, I think if we, right now, our data shows that this methylation was nearly possible. And with these effectors, because I not only combine this DNA demethylase, I also use KTM4C, which is a histone demethylase. So they together, they work really best. And with neurons, because they don't divide more. So I think in neurons, they will work much more better because we don't have a chance for the methylation to come back. But for activation, for example, in those cells, that activation should be directed to there. So I think this combination of VP64 was so much effective. And for Dr. Stormy, and I also wanted to ask that if they could send us our patient-derived fibroblast so we can check these tails in there.
Okay, great.
Yeah.
So a little bit of, okay, great. I was just wondering if you could talk a little bit about your DCAS9 system that you were mentioning as sort of future steps.
Yeah, because in tail is very small. We use is 1.6 kb. So they are good as a final approach. But if we are talking about 6 MB region, this in Prader-Willi IC. So screening for making so many tails in this region is not a very easy task. So with DCAS9, we are also fusing TET1 and trying to make VPR or some other fusions with DCAS9 and try to check. There is a recent paper about how DCAS9 can be used to activate different locus. So we will try to improve their fusions, and let's see if we can make some robust as activator as a tail, and then we will use this DCAS9 platform and make a library of this 6 MB region, and it will be easy to differentiate which regions are more critical and which region are not as much. So hopefully this fusion will be big, because DCAS9 itself is like 3.8 kb, and then if we fuse TET1 and the other fusion, it will be long. So this will not be final, maybe taking into patient, but this will help us to narrow down the region which is more critical in activation.
Okay, great. That sounds very exciting. So Claudio asks, Mussolino's asked, they've done some pilot experiment in post-motorized neurons in vitro, and they could alter DNA methylation patterns. And so that's been done using one of their modifiers by one of their collaborators.
Yep.
Great.
Yeah.
Go ahead, sorry.
Yes, Dr. Claudio was like, he's my supervisor in lab. So he was just explaining that Dr. Strommy's point that yes, we can do, and we hopefully our fusion will be much more capable to activate these cells, post-mitotic cells.
Okay, great. Okay, so it looks like there's no more questions. So with that, we will move on to our next speaker. Thank you so much, Subtain. Our next speaker is Alexandra Hellwach, who's from the University of Edinburgh, and she's going to be talking about the role of SNORD115 and SNORD116 clusters in the development of Prader-Willi syndrome.
Hello, my name is Alexandra Hellwach. I work in the lab of David Tollerby at Wellcome Center of Cell Biology at the University of Edinburgh. So here is a map of the fragment of chromosome 15, so-called Prader-Willi syndrome locus. It is the region that is not functioning properly in people with Prader-Willi syndrome, most frequently due to deletions of many genes in this region. The most common deletions are long, and breakpoints are marked here on the figure. All deleted genes contribute to the patient's phenotype, but not all of them are equally important and causing symptoms. The shortest described genomic deletion concerns so-called SNORD116 cluster, which is marked here with the orange arrow. SNORD116 cluster is a very interesting region. It is maternally imprinted, which means it's active only on chromosome inherited from father, but it's also a source of multiple non-coding RNAs with potential regulatory functions. So here is a simplified map. Like every gene, this gene is used by the cell to make RNA, and there are many different types of RNA that are made from this gene. Some RNAs come from exons, and that can be joined in many different ways and give rise to different long non-coding RNAs. Some RNAs are localized in the introns, and there is 29 non-identical copies of SNRNAs there. And there are also other transcripts recognized here, like SNO long non-coding RNAs and SPA long non-coding RNAs. So deletion of SNORD116 cluster affects all of these transcripts. So this is why we are interested in clusters SNORD116. Why SNORD115? SNORD116 the 5 cluster is part of the same long gene as an HD14 and has similar repetitive structure with multiple SNRNAs. SNRNAs from SNORD115 cluster are highly enriched in neurons, which is meaningful because large part of the Prader-Willi syndrome phenotype is connected with brain development. So from previous studies, also known that SNORD115 and SNORD116 may mutually regulate each other. So as I said previously, we are especially interested in snRNAs. And normal snRNAs, canonical ones, are responsible for chemical modification of other RNAs, usually ribosomal RNA. And there are multiple snRNAs in the cells. They bind the same set of proteins, but they have different sequences and different target sequences. In addition to normal snRNAs, human cells produce many orphan snRNAs without known functions and targets. snRNAs from clusters 115 and 116 are one of those. It's not sure if they form interactions with the same proteins as normal snRNAs, but it's possible that they interact also with unusual proteins and perform non-canonical functions, most likely regulatory. And what is special about them, of course, is their contribution to Prader-Willi syndrome. To study the functions of SNORT116 and SNORT115 cluster, we work on Lumes cells, and they originate from midbrain of developing embryo. Lumes cells grow well in culture, and once induced for differentiation, they create a synchronous population. In 1-2 weeks, they differentiate into electrically active dopaminergic neurons, which makes them a good physiological model. So first, we confirm that Lumes cells are producing our snRNAs. As you see on the northern blot, in undifferentiated cells, there is not much SNORT116, and we don't see any signal from 115. And both kinds increase dramatically with differentiation. So SNORT116 plateaus somewhere between day 6 and day 10, and SNORT115 probably closer to day 15. As I said before, those snRNAs are present in clusters of many non-identical copies, and all of them contribute to the signal on order blot. So to find out which copies of those snRNAs are produced in the cell, we performed small RNA sequencing. Here you have a profile of expression of all 29 copies, and you can see that there is high variation in the expression level. So when we color the plot by the presence of the consensus CD boxes, you can see that the ones that have CD boxes are expressed higher than the ones that don't have CD boxes. So this leads us to the conclusion that snRNAs from those clusters most probably form canonical complexes. Yes, so I used Lumes cells and CRISPR-Cas9 technique, and we created cell lines with deletion of clusters of interest, creating 3 cell lines: wild type, H116, and H115. So with this northern blot, we confirmed lack of expression from deleted clusters. And so we used those cells, wild type and mutant, to determine the role of clusters during neurogenesis. We have performed a time course RNA-seq experiments from day 0 to day 15 to find out which genes are active at which stage of neuronal differentiation. At first, we confirmed the experimental data and looked at the expression from chromosome 15. So what you can clearly see is lack of expression from the deleted clusters, but also worth noticing is the lack of apparent, lack of changes in the neighboring region. So before comparing wild type and mutant cells, let's look at the global changes that happen as cells change into neurons. On the heat map, you can see that most of the changes happen between day 0 and day 6. You can also, it's also confirmed by volcano plots, where the highest number of statistically significant genes is between day 6 and day 0. On the x-axis, there is a difference between wild type and mutant cells, and on the y-axis, there is significance. So, on the contrary, at the end of differentiation, between day and 15, there are hardly any changes in the gene expression. So now let's look at the comparison between wild type and mutant cells. Wild type and mutant cells roughly follow the same pattern of differentiation, which is not surprising because global changes will produce fatal consequences. So here we compare samples by hierarchical clustering, and the differences start to be visible. Undifferentiated cells cluster together. Day 6 also cluster together, but they are slightly farther apart. And at day 10 and day 15, mutant cells and wild-type cells diverge. Here is a volcano plot that represents differential expression analysis of our RNA-seq data. So red are on the red spots represent genes that are significantly different between wild-type and mutant cells, here 115 and 116. And what you can see is that the high number of genes is changing, and the numbers are higher, surely, than what we expected. So we were also interested to see at which stage of differentiation wild-type and mutant cells differ. So what you can see here is the biggest difference is at day 15. So interestingly, this correlates with the amount of snoRNAs that it's low at the beginning and then increases with time. And this pattern of changes it does not correlate with the expression of host gene, SNH14. And also, if you remember, it also doesn't correlate with the general changes during differentiation, because at the end of differentiation, almost all the genes have already established state. So when we only look right now at the genes that are differentially expressed, you can finally see the difference between wild type and mutant cells. However, something here is striking, which is the similarity between those two mutant cell lines. And in fact, when we compare lists of genes that are different between those two mutant cell lines, there is very significant overlap. And here, just as one example, is a MAGEL2 gene that is known to be in the causative of the Schaff-Yang syndrome and contributor to the Prader-Willi syndrome, which is different in the deletion of SNRT116, but not in the deletion of SNRT115. So on this plot, you can look at the functions of genes that are differentially expressed between wild-type and mutant cells. All of those functions are very important for normal neuronal differentiation and maturation, which is axon development, axon guidance, regulation of membrane potential, forming synapses. And then when you look at cellular compartments, you can see that most of the differences are connected with the extracellular matrix of or membranes. So this led us to the conclusion that probably SNRT116 and SNRT115 are important for the communication between cells. So one last thing that I wanted to show you is PCA analysis of the samples. So here those cells are undifferentiated cells, and as you can see, they form a cluster together, as we've seen on hierarchical clustering. Here are they, maybe I will do like this. Yes, they're undifferentiated cells. Here are day 6 cells. They also cluster together, even if they are slightly farther apart. And here are mutant and wild type cells. So this resembles hierarchical clustering. However, what is interesting here is looking at the pattern of changes, the way that those spots move across the plot. So we believe that this reflects the maturation pathways. And it looks like mutant cells are progressing much faster than wild-type cells, probably creating synapses and axons at the time when they are not yet ready to do so. So here are the conclusions. SNART115 and 6 play a role in later stages of neuronal differentiation. They most probably maintain their canonical structure in interaction with proteins. And it seems from our experiments that they are more important for Prader-Willi syndrome than the host gene. Deletion of SNART115 and SNART116 clusters results in high number of differentially expressed genes, and substantial number of the genes is shared between two mutant cell lines. And the last final conclusion is that deletion of SNART116 and SNART115 cluster may affect cell-cell communication. PCA analysis and other preliminary data suggest that deletion of SNART115 and SNART116 cluster affect correct timing of differentiation process. So with this, I would like to thank David Tollerby and people from the Ravy Lab, our collaborators, Emily Ostervale and Jérôme Cavaillé from Center for Integrative Biology in Toulouse, and welcome Trust for funding.
Thanks so much, Alexandra. We have a question from Anthony Isles, and he asks, did your RNA-seq analysis have enough read depth to assess alternate splicing and or RNA editing?
Yes, we had enough, but we didn't go to this kind of analysis because we noticed that the differences are much more significant than the splicing pattern. We can still do it, but it seems that with the changes that we still have right now, enough work to focus on something different. But it was initial plan, so we had like probably about 130 million reads per sample.
Okay, great. Eric Copps asks, what RNA-seq read counting parameters do you use to distinguish individual copies of SNO RNAs? Because yes, as many reads are often discarded as multi-mapping given the high sequence identity between the copies.
Yes, so I tried different ways, but in the end, I decided to go for very old-fashioned BLAST because it keeps all the reads. So I was able to do half manually to see if things are rejected or not. So there is not so many of identical copies. There are sometimes two that are the same and sometimes three. Generally, they have differences, and it's not so difficult to differentiate them.
Okay, great. And his second follow-up question is, are any of the genes in the GEO terms for secretory granules involved in and endoplasmic reticulum involved in stress response?
Honestly, I didn't pay attention to this because there is, of course, lots of different terms. Yes, like taking into account the previous talks, it would be useful to look into this.
Okay, great. And Jérôme Cavaillé asked, did you examine if among the differentially expressed genes, if some of them could be targeted by SNORD115 and or SNORD116 for 2-O-methylations? So in other words, did you look for conserved canonical base pairing potential between SNORD116 and 115 and misexpressed transcripts identified in your SNORD knockout cells?
We didn't look at this yet. This is one of our plans. However, we would like to also do experiments that are looking directly for the interactions. So to recover direct RNA-RNA interaction, and this way try to look for the base pairing and yes. But surely looking for the potential binding sites is a good thing to do.
Great. And I had a question about, so now that I'm really interested in the cell-cell communication and the timing of differentiation. What sort of future experiments do you have planned to further explore that aspect of your findings?
So for now, we are thinking of doing microscopy to look for the formation of synapses, to generally for the morphological changes in cells, and see if there is different timing in appearance of like the mature neurons features.
Okay, great. We have a little bit of time, so if anybody else has any questions and would like to put them in the chat, Okay, we have one from Alvaro Aranda. In the future, would you consider exploring the role of another non-coding RNA cluster that wasn't included in the PWS region?
So we are generally interested in RNAs. So we are hoping that we will be able to just look for new patterns of interactions and new regulation mechanisms. So generally, if we see any kind of, like for example, we are going also to look for binding proteins, for binding proteins that bind RNAs 115 and 116. So generally, if we see any kind of patterns there, we will try to expand possibly to other RNAs.
Okay, great. We'll just wait a little bit to see if another question comes in. But in the meantime, thank you very much for your presentation. Okay, it looks like there are no other questions, so we'll go ahead and move on to the next speaker. Our next speaker, we have Monika Sledziowska from the University of Birmingham, and she's going to be working on non-coding RNAs associated with Prader-Willi syndrome, regulate transcription of neurodevelopmental genes in human-induced pluripotent stem cells.
Hello, my name is Monika. I'm from University of Birmingham, and today I'll be telling you about the role of long non-coding RNAs in Prader-Willi syndrome, and specifically in how they regulate transcription of neurodevelopmental genes. So to start with, I'm not going to talk very much about the Prader-Willi syndrome symptomology because I believe that you are all very well aware of it. However, let me just remind you that Prader-Willi syndrome is caused by the absence of paternally expressed imprinted genes at the locus 15q11. So here you can see the locus itself, and then in black you can see genes that are not imprinted, and then in blue you can see the paternally imprinted genes and transcripts. And you can also see in red the one gene that is maternally expressed. In particular, this project is concerned with the role of long non-coding RNAs. So in the zoomed-in version of this graph, you can see the portion of the locus where a lot of the non-coding transcript originates from. So you can see that here the SNORD RNAs are transcribed from, that I'm sure you're very familiar with. And you can also see several different cases where Prader-Willi syndrome symptomology is caused by really minimal deletions, which only encompass the part of the region which is associated with non-coding RNAs. This kind of indicates that if you can see the symptoms by only deleting a portion of the locus which is associated with non-coding transcripts, it is worth investigating their role. So let me introduce you to particular species of long non-coding RNAs that this project was concerned with. So bear with me here. This is just a portion of the locus that I've mentioned before, and it gives rise to a host RNA that is then processed into a number of different RNA species. So let's start by introducing the small nuclear RNAs. These are only 60 to 300 nucleotides long. They are synthesized by polymerase II, and they do not have a poly-A tail. However, they form these interesting secondary structures that are called the CD box or the HACH box. What is interesting about these is that they are very resistant to degradation. And what can happen is if within one intron you get two small nuclear RNAs, as they are transcribed and processed, they will form these secondary structures and they will resist degradation. The interesting thing is, when you have two of these, what can happen is that the portion between them, the intervening region, can also resist degradation. As a result of this, we can get something called the small nucleolar long non-coding RNAs, and this project is partially concerned with them. Another interesting thing that can happen in RNA processing is within one intron, you have the small nuclear RNA as well as a PAS sequence. So similar to what we discussed previously, you can get a transcript which on one end has a small nuclear RNA as previously, but on the other end, it's got a poly-A tail. And as such, again, it is resistant to degradation because of the small nuclear RNA and the poly-A tail. So the long intervening sequence in between is also not degraded. So in the Prader-Willi locus, you got 2 SPA long non-coding RNAs, so the ones with the poly-A tail, as well as 5 with the small nuclear ends on both ends. So the role of these transcripts in Prader-Willi syndrome is not well investigated yet. So the aims of this project was to look at the changes in nascent and steady-state transcription following depletions of these species of long non-coding RNAs. We also aim to identify some gene targets that might be regulated by these long non-coding RNAs in this locus. So first, we wanted to find a suitable model for this project. So we have tested the expression of the long non-coding RNAs of interest in a number of different human cell lines. So we checked HeLa cells and HEK cells, which are quite kind of classic models. However, the expression levels were extremely low. So then we have turned to human iPS cells, and there we saw a steady and actually quite high expression of these long non-coding RNAs. So this is the model that we eventually went with. So let me just show you these. These are reads from total RNA or chromatin-associated RNA on chromosome 15. And you can see that the signal for the host RNA that these long non-coding RNAs originate from is quite strong. And in particular, in the kind of zoomed-in version, you can see that it is mostly the number 3 and number 4 small nuclear RNAs that are responsible for this signal. This is just a comparison to some other highly abundant transcripts, and you can see that the small nuclear RNAs are indeed very highly expressed. So the next thing that we wanted to do is we wanted to determine the effect of knocking down these long non-coding RNAs. So in order to achieve this, we have used a system of antisense oligonucleotides. So the way they work is you design a sequence that will target your particular transcript. And then once you introduce that into the cell, it will have the capacity to bind your transcript and trigger degradation via RNase H pathway. So we have designed these antisense oligonucleotides for all of our long non-coding RNAs of interest. Now, we didn't particularly want to investigate each individual long non-coding RNA, as they are often transcribed from the same host RNA, and usually they come together. So we have designed these 4 conditions to look at. First, of course, negative control. And then afterwards, we try to target only the SPA long non-coding RNAs, which there are, just to remind you, 2 of, and then only the small nuclear long non-coding RNAs. And then finally, perhaps the most interesting condition, the one where we have targeted both the SPA and the small nuclear long non-coding RNAs. So firstly, just to have a look at the knockdowns that we have achieved, you can see that in the condition where we have tried to knock down all of the long non-coding RNAs, you see a reliable decrease across the board. When we try to target only the small nuclear RNA, you can see that indeed we see a reliable knockdown of those species of RNA, but also of the SPA long non-coding RNA too. And this is because they are transcribed and then processed from the same host RNA, so there will always be a little bit of overlap. Then in terms of the SPA long non-coding RNA, again, perhaps not so reliable, and you also get quite a lot of knockdown of the small nuclear RNAs. So firstly, we had a look at total RNA, where upon the knockdown, we have extracted the total RNA and we have sequenced it and then tried to identify some differentially expressed genes. So here you can see that the changes, they were very subtle. So even with a knockdown of all RNA species, we only identified a couple of differentially expressed genes. Something that has been tried before. So in order to try something perhaps a little bit new, we looked at RNAs that are associated with chromatin. So these are primarily the transcripts that are being actively transcribed, or you can also refer to this as nascent RNA. So in order to achieve, in order to extract chromatin-associated RNA, we use fractionation. And following fractionation, we again sequenced our samples. So here you can just see the reads for the different conditions, and you can see that we have achieved quite a good knockdown, especially in the condition where we have targeted all of the long non-coding RNAs. But the truly interesting result comes from the analysis of differentially expressed genes. So you can see here, firstly, in the two conditions which only targeted either the small nucleolar long non-coding RNAs or the SPA long non-coding RNAs, that again, the observed differences were quite small, although you will also note that there's a couple of potentially interesting genes in the small nuclear RNA condition, which are Neurexin-1 and FAT3. However, we got quite a lot of hits in the condition where we have knocked down all of the long non-coding RNAs, which was really quite promising. So let me just talk you through some of the genes that we have identified. So firstly, let's have a look at some of the downregulated genes. And what we have found is that the three top downregulated genes were Neurexin-1, Neurolegin-1, and FAT3. And here you can see the kind of dot plots showing the counts associated with these two genes. And then you can also see the reads where you can see how across the gene you can see dampening of the signal. So the reason why these are quite interesting is because they're all are known to play a role in neurodevelopment. Specifically, Neurexin-1 and Neurolegin-1 are synaptic proteins, they're adhesion proteins. They can form heterodimers across the synapse, helping with scaffolding of the cell, and they also help during the development of the synapse in terms of gathering of the necessary proteins and creating a scaffold for the synapse to form. Similarly, FAT3 is a cadherin that also plays a role in synaptic adhesion. Then in terms of the genes that were upregulated, I have to say that the effects here were a lot smaller, and there were fewer of these. However, there was a few potentially interesting hits, primarily genes that were associated with cell metabolism and cell death. So one question that you might ask yourself is how come we are seeing neuronal markers in what effectively is a stem cell line? So I was wondering about this as well. And what I tried to kind of investigate is I had a look at different markers associated with different cell types in our samples of total RNA and chromatin RNA. And as expected, we have found a great abundance of proliferation markers, which is something that you would expect in stem cells. But we have also found some expression of neuronal markers. However, it is worth noting that it is primarily neuronal markers, but not markers of functional neurons, which would be indicative perhaps of the fact that the cell, even in a stem cell form, might be transcribing some of these genes and generating some of these transcripts in preparation for differentiation. But as they are not used, they are then degraded. And this is kind of supported by the fact that we see a lot more of these in the chromatin RNA rather than in the total RNA fraction. So we should also have a little look at the geoterms that were associated with the upregulated and downregulated genes in these samples. So kind of in line with the genes that we've already discussed, in downregulated geoterms, we see a lot of cell adhesion and synaptic function, which is really quite interesting in terms of the symptomology of Prader-Willi syndrome that we see. And then again, in terms of the upregulated genes, we see quite a lot of metabolic processes. However, again, it is worth noting that the effect sizes of the upregulated genes are a lot smaller. So finally, we have performed this analysis called the GSAA analysis in order to identify small coordinated changes that might be present in the data set, but that are not obvious from just looking at the differentially expressed genes. So again here, we have identified some pathways that might be affected, and we see, as expected, central nervous system development, as previously. However, let me draw your attention to the fact that there is also changes in the immune system pathways, which again is quite interesting in the context of the literature around Prader-Willi syndrome. So in conclusion, as a result of this one-year project, we have generated some preliminary data which could be potentially interesting and that we'll explore further. So we have found that knocking down the long non-coding RNAs that originate from Prader-Willi locus can affect transcription in human iPS cells. And specifically, knocking down of these RNA species can result in downregulation of transcription of neuronal genes, which again can be quite interesting in terms of the symptomology of Prader-Willi syndrome. So thank you kindly for listening, and thank you to Grzechnik Lab and Garcia Lab and Dan's Lab.
Thank you so much, Monica. We're going to start off with kind of a question and a comment. So Larry Ryder says you may need to differentiate your iPSCs into neurons first, and then you may get more hits. And then a similar question is, have you tried testing knockdown in different stages of neuronal differentiation, or does that impact the ability to differentiate the cells?
These are very interesting questions, and indeed the ones that we ask ourselves. So this is the next stage of our project for the next year, which we just started. We are in the process of differentiating our cells, and we'll be knocking down the species of RNAs at different stages. And we will also be looking at changes in the chromatin associated as well as total RNA at these different stages of differentiation. So yes, I absolutely agree.
Okay, great. Well, that'll be very interesting results. We look forward to hearing more about that. Rob Nichols asks, have you used independent biological samples and quantitative RNA analysis and Western analysis to confirm your RNA-seq changes?
Right. So the first question is independent samples. So reasonably, yes. So what we have done is we use the same cell line, but we have done the transfections several times at different stages, and they all kind of show the same thing. We haven't done any Westerns yet. That is one of the things that we were planning to do. However, I do not have the results for you yet.
Okay, great. And another comment from Larry Ryder, he says stem cells typically express many genes that are later regulated differently in differentiated neurons. So your upcoming results, or your upcoming experiments, I think. Jim Resnick asks, are SNO RNA expressions unchanged in the knockdowns?
SNO RNAs. Yes, so I think we did look into this, and I don't think we have found any changes of SNO RNAs as far as I remember. I remember we looked at it using qPCR.
Okay, great. Jerome Caval asks, are the SPA long non-coding RNAs and or the SNO long non-coding RNAs evolutionary conserved in placental mammals? And if so, can they be detected in vivo like in humans or mice?
Okay, so as far as I remember, the SPA1, one of them is conserved in mice. However, the SPA2 is not. Whether they are detectable in placenta, I don't know. I'm not sure whether that has been checked.
Okay, great. Oh yeah, and can they be detected in the human or mouse brains? I guess was the.
I don't think this has been investigated, because these species of RNAs have been described quite recently. And I think, as far as I know, all of the work on them has been done in different cell lines. I think both mouse and human cell lines. However, I don't think we have tried yet to see them in the brain, which of course would be extremely interesting.
Okay, great. Another question, have you checked how the knockdown of long non-coding RNAs affect the levels of SNHG14?
Yes, so there seems to be a decrease in the host RNA upon the knockdown.
Okay. And then another question, were you able to detect SNO RNAs in the libraries in order to quantify them with certainty?
Yes, as far as I remember, yes.
Okay, great. So I know you mentioned a little bit about your kind of future experiments, but now that you kind of know about the downregulation of your transcriptions, what are your next steps with the next year of the project?
So the next year of the project will be particularly focusing on the differentiation and then knocking down at different stages of differentiation to see whether there are changes on transcription as well as on differentiation itself, as we have heard before from Alexandra. This could be an interesting avenue. Another thing that we wanted to do is try and confirm the mechanism as to how these long non-coding RNAs impact on transcription. And one possibility is that this happens via binding certain RNA binding proteins. And from previous literature, we have some kind of indication of what these might be. So we will try and confirm that as well to give a little bit of a mechanistic spin to this story.
Okay, great. Well, thank you so much. We will move on to our last speaker of the day. Next, we have Chi Wu from Baylor College of Medicine, and he's going to be discussing the reciprocal control of obesity and anxiety depression disorder via a GABA and serotonin neural circuit.
Hello, this is Chi Wu. I'm assistant professor from Baylor College of Medicine and also working in the associate with CNRC of the U.S. Department of Agriculture. My lab actually interested in the neurocircuitry control of the metabolism and feeding behaviors and a variety of other neurological diseases, such as like this Prader-Willi syndrome. So I would like to take this opportunity to present an exciting project we recently published about reciprocal control of the obesity and mental disorders where the GABA serotonergic neurocircuitry in the brain. As we have been known for many years, the metabolic syndromes like obesity and diabetes are heavily being comorbid with a variety of the neurological disease, especially the mood disorder and anxiety disorders. So clinical data suggesting that the body weight are in the strictly a positive association with the depression and anxiety levels in the human patients. As of how this thing has been working in the brain, there's very poorly understood as of the neurocircuitries and the molecular substrate in the brain. So our lab has been studying the AGRP neurocircuitry over the last about 10 years, that especially the neurocircuitry controlling about a variety of the feedings and other functions. So this is like an overview about a few years ago published by the review that how complexity of the AGRP neurocircuitries interacting with a variety of the deep brain tissues outside and inside of the hypothalamus, and all the way as far as in the brain stem or even in the thalamus or even the cortex. So the AGRP neurons are very small population located in the very bottom of the arcuate nucleus of hypothalamus, but has a prominent role that has been identified many years ago regarding how they promote appetite and suppressing the metabolism. In recent years, that has been multifunctionally identified by my lab and many other labs that they participated in the control regulation of a variety of the critical neurological and physiological functions. So taking some examples by using this diagram that we show recently that a small population of the AGRP GABA neurons that are sending projections to a peribrachial nucleus in the brain stem that are regulating the consummatory food intake. And meanwhile, there's a group of the dopaminergic neurons that project not to the other exclusively to the brain stem, the same neurons that are regulating motivational feedings and suppress the food satiation, and indicating that they are also heavily involved into the feeding and foraging behaviors. And also, meanwhile, we find a unique group of AGRP neurons regulating energy metabolism and thermogenesis, and that also involved in obesity and anorexia functions. And in the most exciting and most recent study done by my lab is to identify another group of AGRP neurons that sending projections from the hypothalamus to the BNST in regulation of the comorbidity of obesity and the mental disorders through GABA and the serotonin interactive manners. So I would like to take the next of the times to discussing this exciting project. So what exactly, the main question is, what exactly the neurocircuitry and the signaling mechanisms underlying this, such a comorbidity between the obesity and the complex mental dysfunctions? So as we have known in both animal models and in patients that the high-fat diet, chronic treatment of high-fat diet will cause severe problems, not only to increase the body weight and metabolic dysfunctions, but over the times, they're actually causing the comorbid mental problems. So in the animal models shown here is an open field test that after 6 weeks of high-fat diet exposure, the animals are showing severe anxious-like behavior that they actually avoid the center part of the open field test. And similarly, using this elevated plus MACE test, we show here that after high-fat diet treatment, the animals are avoiding the open arms as compared to the lean mice. So this gives us some hints that animals are behaving in similar kind of patterns, just like humans, when they develop this comorbid high-fat diet-induced obesity and metabolic symptom. And also, we want to using this new emerging technologies called in vivo calcium imaging platforms to real-time monitoring a specific group of HGF neurons and downstream neurons, how they interact with the high-fat diet and during this movement in the anxiety, depression, behavior paradigms. So in order to achieve this, we cross HGRP mice with the ROSA GCAM6 mice to specifically expressing these calcium sensors into the HGRP neurons in the hypothalamus. And we can able to recording their neuronal activities in the in vivo kind of situation. So first of all, showing here is a low-fat diet treatment. You can see that this is a baseline level of the HGRP firings. And as long as you perform this fasting, the HGF neuron should be hyperactivated as expected because these neurons are promoting appetite. And after chronic treatment of high-fat diet, interestingly, this HGF neuron has been desensitized and eventually losing this fasting-induced hyperexcitabilities, as shown in the last rows. So so also we want to testing that what exactly the HGF neurons are behave during the anxiety, depression, behavior paradigms. And in the lean and healthy mice, the HGF neuron firings are more in the center point when they are perform this open field test. After showing this is the calcium imaging real-time patterns over like 30 minutes patterns, and the correlation between them is very high. And after give a chronic exposure to the high-fat diet, the HGF neurons has been severely desensitized, and the mice are showing much less activities in the center part of the open field, indicating they have a correlation between this HGF neuron firing desensitization and the avoidance of the center part, which is indication of the anxious response. And also want to show you some data here, which is the videos that can directly showing that how this HGF neuron firings are positive correlate to the center part in the lean and healthy mice, and especially when they're in the centerpiece of this open field test. And in contrast, if those high-fat diet fat mice are severely anxious, and they are most of the time they avoid in the center part, and their HGF firings are proportionally diminished significantly. And this is a statistical data showing this difference. So we're using, in the next step is using the optogenetic and the chemogenetic approach, trying to manipulate the HGRP projections from the HGRP to the one of the critical targets is dorsal BNST, and identify how they involved into this regulation. As you can see that the HGF neuron will be activated when they are the projection to the BNS in terms of promoting the food intake. And meanwhile, the elevated plus MACE test showing that these mice are activation of the HGF neurons are showing this more anxiolytic response, and same like the open field test they're showing the anxiety when HIP projection to the dorsal BNST has been activated. And in the chronic or chemogenetic test, we're testing both suppression and activation of the HIP projections to the dorsal BNST. And on the left side is the DQ expressed HIP neurons that when given the C and no inducers, the mice were showing the similar response that actually they're showing anxiolytic effects. But if you inhibit the HIP neuron projection to the dorsal BNST, the mice showing the exactly opposite and like anxious and more anxious behaviors. And similarly, the open field test indicating that the same kind of a response for separation and activation of this HIP projection to the dorsal BNST. So the next step is we're trying to figure out what's the molecular substrate or significance underlying this regulation of the HIP projection to the DBNST in the suppression in anxiety depression regulations. So we find out that there is drugs that have severe which is zonisamide is an FDA approved drug that has been known to have a significant side effect of regulating body weight while they're actually using them for treating those psychiatric diseases in the human beings. And these drugs has been shown here when they chronically infused into the dorsal BNST of the MC4 neuron expression. MC4 Cree mice express the calcium sensors in these neurons. We are able to record them in the before and after we give them the high-fat diet treatment and after the treatment of this zonisamide into the BNST. And showing here is that high-fat diet treatment will actually significantly activate this group of MC4 neuron in the BNST, and the zonisamide treatment can significantly suppress them, the firing patterns. And behaviorally, we show that the treatment of the zonisamide can suppress food intake significantly, and as well as their anxious depression levels has been significantly depressed. Furthermore, we actually using a variety of approach, including this calcium imaging approach that to identify there's a group of the GABA-A receptors participating into this regulation of the anxiety depression over this MC4 neurons. And of particular, the alpha-5 subunit of the GABA-A receptor is critical for participating in regulation of the mediating this zonisamide-mediated suppression of anxiety depression. And data showing here is that the GABA-A5 knockout mice, which is the GABA-A5 sgRNA mice, which is showing that the significant hyperexcitabilities of the MC4 neurons because they're losing a significant portion of the GABA-A signaling input. And interestingly, that by giving the zonisamide into those GABA-A5 knockout mice, the excitabilities of the MC4 neurons is failed to be suppressed. So indicating that the alpha-5 subunit is important for mediating of this zonisamide-induced anxiety depression function. Furthermore, we find these GABA-A receptor alpha-5 subunits are co-localized with another group of the serotonin 3 receptors in the same group of the MC4 neurons in the dorsal BNST. And we're trying to figure out how they are working together in the positive and negative ways. So first of all, we give delete the GABA alpha-5 subunit, and as expected, showing in the middle of the column that this mice actually showing more anxious behavior. Interestingly, if we simultaneously delete this alpha-5 and serotonin 3 receptors, we achieved rescuing effects that this mice anxious level has been restored into normal, as shown here, the quantified results. And using the elevated plus maze, we also achieved a similar kind of conclusion that the deletion of the alpha-5 GABA-A receptor induced anxiety depression response can be rescued by simultaneously inactivation of the serotonin 3 receptors, as shown here. And behaviorally, interestingly, that the in the metabolic aspect, that the GABA A receptors and the serotonin 3 receptors are participating also coherently to regulating obesities over the long term. So here is showing that a high-fat diet treated mice has been simultaneously given a combination treatment of the dolutamide and the granisetron, which is a selective serotonin 3 receptor antagonist, infused into the dorsal BNST or systemically through the IP injection. We find that over the chronic treatment of such the combination of drugs, that the feedings and the body weight have achieved a significant restoration into the healthier level as compared to the mice in the lean stage. So this is basically showing that this pathway is simultaneously regulating the anxiety, depression, and the comorbid obesity symptom. Interestingly, we also identified their feeding behavior changes after with co-treatment of the zolpidem and the granisetrons targeting these two pathways simultaneously. And interestingly, this is a food preference assay showing here on the left side of the panels, which is the mice chronically being given the low-fat diet, and then performing this food preference test, they have sort of a craving of the high-fat diet as expected. And after like 6 weeks of exposure in the high-fat diet, those mice are displayed even more severe craving of the high-fat diet, and their overall calorie intake has been also further enhanced, about 20 to 30%. We provide these drugs to them as chronically over the 2 months period. We show that we show a dramatic reduction of the total calorie intake, and interestingly, a dramatic percentage reduction of the high-fat diet intake over that total calorie intake. And we achieve this kind of the goals by both IP injection of the combination drug and or intra-BNST injection of the drugs. And this is a sort of dissections of the nutrient levels of the intake of the food. They are showing here that the fat content of the food during this food preference test has been dramatically reduced, while this healthy carbohydrate and protein level has been compensated for the loss of the reduction of the fat in consumption. So indicating that this is a very interesting voluntary switch of the diet preference from high-fat diet to low-fat diet by using this combination of the two FDA-approved drugs in vivo and during the in vivo test. So the take-home message of this study is we find a particular group of AGIP neurons that send GABAergic projections to the MC4 neuron located in dorsal BNST, and which is expressed in that the alpha-5 GABA A receptor and serotonin receptors, and the achieve the in the healthy situations that the GABA inhibitory input and the excitatory input of the serotonin achieve the balanced ways to regulating the mental levels and the food intake, the appetite. During the sort of a chronic exposure to the high-fat diet, the AGIP neuron has been chronically desensitized, causing the hyperexcitation of the downstream MC4 neurons and causing the variety of the issues. If we're able to achieve by re-establish the balancing to give this simultaneously, the dolutamide and the granisetron treatment to re-establish the balanced sort of a neurocircuitry input within the dorsal BNST, and ultimately achieving our normal mental state and healthier body weight. So this is sort of a first-hand hint showing that the comorbidity of such a complex neuroendocrine and the neurological disease can be understandable by dissecting this particular one or more neurocircuitry and the key signaling mechanisms. Lastly, I want to acknowledge the first following personnel, that especially the postdoc and the student from my lab, especially Dr. Han and Dr. Shar, which is a postdoc from my lab, and a few other people that past associated in my lab, and a variety of the collaborators that inside the Baylor College of Medicine and in different kind of places. So thank you so much. I will take the rest of time for more questions. Thank you.
Thank you so much, Chi. As a reminder, if anybody has any questions, please put them in the chat. This is very exciting results, and I think has a lot of application, moving forward. So what are your next steps and what are your next experiments after seeing all these results? I think you're muted.
Okay, so our next step is trying to further explore the deeper brain regions that with functional connectivities with this newly identified neural circuits in the regulation of the comorbidity of obesity and the variety of the mood and mental disorders. And also there is sort of a functional study about novel drug targets within this individually differentiated subpopulation of neurons in this neural circuit. Thank you.
Okay, great. Do you think, so when do you think sort of the human aspect will come in, or what else do you need to do in the animal model?
Yes, I think that this is just like the beginnings of our understanding about the potential, high potential translational kind of interest into how to sort of tackle this difficult sort of metabolic and mental disease. And we believe that there's sort of a future sort of translational sort of efforts needs to be put onto this newly identified circuits, and these two separate class of the compound, and studying their sort of a combinatorial sort of efficacy in tackling this disease, and also further other sort of novel drug targets in the future.
Right, exactly. So do you think this will translate into screening patients like for purposes of personalized care when you're seeing sort of the obesity and mental health issues?
Yes, I think that definitely have a lot of those implications. So we believe that the current knowledge and potentially few sort of can fuel this sort of the clinical efforts to do such kind of the screenings, and which I believe that's going to potentially have profound sort of beneficial effects. Yeah.
Great. I think we have time for one more question. So Shuang's asks, have you looked at other 5-HT antagonists like 1A and 2A subtypes?
Not at this moment, but we are highly interested, providing that the genetic mouse models support this sort of directions in the future.
Okay, great. Well, thank you so much, Chi, and thank you to all of our speakers today. A special thanks to all of our sponsors who are named at the top of our landing page. And of course, thank you so much to all of our viewers for joining us to hear about all of this exciting research. So a few things, please, if you haven't yet, please check out the poster session. You can find the links to view both the PDFs of the posters and the pre-recorded poster walkthroughs at the bottom of the event landing page. So we look forward to seeing you here again tomorrow. Please note the symposium is going to start 30 minutes earlier tomorrow. So it will start at 11:30 a.m. Eastern time. So thank you so much for joining. This concludes day one, and we will see you back here tomorrow.